good day to distinguished guests, ladies and gentlemen. Welcome to our webinar sponsored by Shandong LachanceGroup with the theme of changes and challenges in aquaculture nutrition. So my name is Benjamin. I'm your host for today's webinar. So before we start, please remind with our housekeeping rules. All the microphones and videos of the attendees are automatically muted. And if you have any questions, please type your question in the Q&A box, and we will entertain them after each presentation of the speakers. So should there be any disruption during the webinar, please be patient and try to sign in again. So, and a gentle reminder to all the speakers, 45 minutes for each presentation. And after that, 10 minutes for Q&A. So total will be 55 minutes for each presentation. And so now, without further ado, I would like to introduce our first speaker. She is Director of Feed Technology. She is Director of Feed Technology Innovation Team from Institute of Feed Research of Chinese Academy of Agriculture Science. Her research area involves the basic research of feed processing, research on improvement and innovation of feed processing technology, and development and application of new protein sources such as cottonseed protein concentrate, single-cell protein in animal feed. Also, she has published more than 130 papers. Ladies and gentlemen, please welcome Professor Xue Min present her topic. Bile acid metabolism pathway regulate metabolic hepatic disease in fish with various trophic level induced by multi-factors. So it's very long title. So Professor Xue, now is your time. Please.
Thank you. Do I need to open my video or not? Can you see my screen?
Yes, yes.
Okay, thank you. Thank you, Dr. Bai. Thank you, LachanceGroup, invite me here to give the presentation. Yes, it's quite long in my topic because we have doing some works on the bile acid metabolism in the fish for several carnivore species and also in common carp. So today I will introduce my work on the following 4 topics: how plant protein and starch level in carnivore species, and some works on the bile acid intervention, and I will give some future work in aquaculture for crosstalk between the bile acid metabolism and microbiota. So the first wall, I will introduce our work for 12 years for alternative protein in diet of marine species, spotted sea bass. As early as in 2010, we have conducted the first trial in this species. We replaced the fish meal by the fermented soybean meal to replace the 25, 50, 75, and one more group for 100 group, and totally replace the diet for 60 weeks. And we have found some interesting work, and we repeat this work in 2013, 2017, 2018, 2019, and in last year. So we continue to do this work and found very interesting results. As in this slide, you can see in the first trial, we can find the feed intake was suddenly reduced with a higher level of fermented soybean meal, and interesting phenotype. We find that in the the egg works, after the feeding for egg works, and this group, the total fish, plant protein diet, the fish have been died with a very high mortality. And we keep these 3 groups, but we found that the fish feed the diet with the 50% replacement had an even higher feed intake. And when we continually conducting this trial, finally find a very small difference between the groups. It's not the same as the first of the during the first 8 weeks. So the feed intake during this time will be the most important issue for the strategy to deal with for the high level of fish, the plant protein. So we repeat the diet and find that for short term, during the first 3 weeks, the feed intake was very significantly reduced for 73%, and the growth was also have negative growth during this time. And we found that in this trial, the growth was also still reduced following the higher level of plant protein. And then in the the third trial, we confirmed the phenotype for the adaptation and the higher level of feed intake of the fish when feed high level of plant protein. And this adaptation mechanism was related to some gene and the protein expression of the TOR pathway. And we tried to use some attractants to make up the feed intake, and also the umami can give the higher level of feed intake. So in the 2029, very interesting, when we repeat this trial, and we found after almost 10 years feeding for this species, the feed intake is not very important issues, and the plant protein diet and the fish meal have no difference on feed intake. But in this trial, I will give the case will be the further explanation. So we compare the high fish meal and high plant protein diet, and we balance the essential amino acid and the phosphorus lipid for fatty acids. And in these 2 groups, even for the plant protein groups, and we found the FCR is 1.11, and it's higher than the fish meal, but it still can be accepted. But we found the feed intake is higher, so we more focus on the to find some problem. It's okay for fish health, but in this group we find significantly higher hepatic somatic index. So we further determine the amino acid metabolism in the plasma, and even we balance the essential amino acid by the crystallized amino acid, but we still can find that for the lysine, serine, valine, and these 5 amino acids still in the status of deficiency, and the arginine, the histidine is higher than the fish meal group. So the amino acid balance is still the problem for when we use the plant protein to replace the fish meal. So we found the significant reduced in the plasma for essential amino acid, but higher non-essential amino acid in this group. For the glucose metabolism, and we take the samples of 24-hour postprandial. It's at the status of starvation, but we still find very high expression for the glycolysis and glucose aerobic oxidation. That means the lysis is still the very high level of expression. the lysis and makes the basal metabolism will be higher. Sorry. Oh yeah. And for the lipid metabolism, and although the lipogenesis and the lipolysis beta oxidation, both of them are higher level of mRNA level, but the lipolysis and the beta oxidation is much higher than the lipogenesis. So this also means the lipid metabolic rate in plant protein group is higher. And for the culture and the metabolism and bile acid metabolism, and for the phenotype, the plasma culture is lower in plant protein diet group, but in liver we find the phenotype is the culture is much higher than the Fishmeal group, and so the fatty liver phenotype is mainly also because of the cholesterol accumulation. And although in plant protein it's deficiency in the cholesterol, but the fish can synthesize the cholesterol by themselves. But the cholesterol synthesis didn't go to the bile acid synthesis pathway, but the higher level of cholesterol was accumulated in the liver. So we determined the basal energy metabolism, and yes, we found the T3, T4, the hormone level plasma is much higher, and the second messenger CAMP and CRAB is for the biomarkers for energy metabolism, both of them are higher. So in this group, the higher lysis phenotype will be induced the inflammation and immunology stimulation. So we further determine the phenotypes for the histology of the liver. And in this plant protein group, we found much more the samples with the fatty liver and with the higher expression for the P65NF kappa B, and the cleaved history for the apoptosis biomarkers. So, but we didn't find the JNK phosphorylation still. That means, so we give a summary of this trial that when we face a high level protein, high plant protein diet, and that will be induced the systematically metabolic disorders that including glucose, lipid, and bile acid, energy, and so on, amino acid, all the nutrient metabolic disorder was observed, and the fatty liver mainly induced by the cholesterol accumulation, and this makes the fish in the high expression with the TNF-alpha. This is the inflammatory cytokine and induced P65 activation, but the JNK phosphorylation is not activated. So this data still at the phenotype. Maybe when we give the medicines or give some interventions, will be cure. Maybe give some clinical strategies will be resolve the problem. So in the in last year, we gave the intervention strategies, repeated this trial to we replaced the Fishmeal by the SPC. So we also gave the HGCA from the long-term group to in this trial, we also find the phenotypes of the fish when we give the high level of plant protein SPC, the fatty liver, and some phenotype of microvascularizations phenotype was observed in this group. And also this fish, even we didn't give the cholesterol in the diet, but the spotted sea bass can synthesis the cholesterol itself. Even we give the bile acid, the cholesterol synthesis is not give the negative response. It's even higher expression with the cholesterol in the liver. And another interesting phenotype is we found in the when we give the intervention of HDCA, this group will be in the liver, in the liver and in the bile, gallbladder, and will be observed a higher level, a higher content of glycol-taurine bile acid. That's it. Conjuncted bile acid will be improves health of the fish that have been observed in other studies. So I give the summary of the spotted sea bass is in the 12 years study, we found that the early feeding adaptation was observed when we feed the full plant protein diet, but the lower growth performance are still observed. And the main cause of fatty liver is disorder of glucose, metabolism, and lipid, and increase the cholesterol compensatory synthesis, but inadequate bile acid synthesis. So we also observed the breaking the break of the P65 and JNK pathway on the TNF-alpha induced apoptosis. The hypothesis on the function of glycine conjunctive bile acid in elevation of fatty liver of spotted. So we still not confirmed this hypothesis still. We have interest into further study on these topics. The second trial species is on carnivorous sturgeon, a marsh sturgeon. So this is one of the most important sturgeon species for hybridization. It's generally with hybrid with the Siberian sturgeon in the aquaculture, and all sturgeons was in the protection list of the CITES. So this is a high trophic level species, but we still have a lot of information on this species of their nutrition requirements. In this trial, we generally use the same formulation of the sea bass. We have a 3 diet with the high fish meal, and we replace the fish meal with 50% and 100%. In the full plant protein diet, we found the high level of mortalities and the lower growth performance, but we find the higher feed intake in this species. It's very interesting. And of course, in this group, we didn't find a higher hepatic somatic index, but these two groups with a smaller size of liver. We firstly find very serious damage in the spiral vessel intestine. It's a very strange structure of this sturgeons. The sturgeon have a very strong intestine with the spiral valve, and that will be very high error. It starts with the time. And when we use the histology analysis, we found even in the 50% replace mild damage was observed, and in the full plant protein, very serious damage were observed in this group. And we also observed the conjunction, tight conjunction gene related to gene expression was significantly reduced. And also we found the serious necrosis in tissue in the intestine was observed in the high protein plant protein diet. And we also find some microvascularization phenotypes in the fish with the high level of plant protein. The microvascularization is a kind of a phenotype with it's a kind of pre-fibrosis. So when liver needs more nutrients, and they will have more proliferation of the collagen structure of vascular to make sure the higher level of blood flux can be transfer more nutrients to the liver tissues. So we also find in these two groups have a higher level of lipid in the structure of liver. In this trial, we found very serious phenotype like the aging, or it's quite similar to the mammals with the cancers, something like that. The phenotypes, or not only with the lower expression of the tight conjunction, but also we found the lower expression of the antioxidant gene expression and in the liver and in the intestine, and also for the microbiota in the intestine, and the Clostridium is significantly increased in the group, as could be this group of bacteria will be induced some reaction of the gas production, something like the gas quite be harmful to the tissues, and so the microbiota it's not in the kind but in the tissues of the intestine, and also related to immunology response of the fish. In the liver, and we found in this species higher level of culture synthesis gene increased, and so no problem for the sturgeon can synthesize the cholesterol, and also for the bile acid synthesis gene also improved. So we suppose the liver of among sturgeon, they can synthesize from the cholesterol to the bile acid, but we found in the intestine and the transfer bile acid transfer gene and the receptors like FXR and the TGR5 are both of them are significantly downregulated in the high plant protein diet. So the hypersensitivity is for the sturgeon, the phase 1 intake high level of fish plant protein, the synthesis of culture to bile acid is no problem, but the transfer and the receptors will be downregulated. So we also find the lower autophagy of the proliferation in tissues and the lower protein of ERK. So all these also biomarkers for the cell proliferations. So I give the summary of the sturgeon is that adaptation of 27% of plant protein will be not okay for the fish, but high level of plant protein will disturb the intestinal microbiota and the risk of cause fat accumulation and the prefibrosis in the liver. And the full plant protein diet destroys the mucosa of the valve intestine and lead to the oxidation stress in the intestine and the liver, which will in turn cause the tissue immunology suppressions. And for the bile acid reabsorption transportation will lead to the hepatic cholestasis. And the intestinal and the liver immunity, autophagy, and others repairability is very low, as eventually leading to the death of our sturgeon. So for this species, we have interesting to study how the intervention of bile acid supplementation in feed will be interesting for us to further verify. And I will give the second part is the high starch diet induced metabolism diet, especially for the largemouth bass. This species is very sensitive to the starch, and it's as early study of 2014 when we give the fish for the artificial diet or the chili fish, we compared the fish with the artificial diet with 17% of starch, will be induced very serious cholestasis phenotypes in this fish that accordingly induced a high level of TBA and AKP in the plasma. This is the biomarkers for cholestasis in clinical studies, and we found the serious necrosis, apoptosis, and inflammatory responses, and a high level of fatty acid synthesis and fatty liver phenotypes. And accordingly, we conducted a trial with large mouse bass. We compared the 9% starch and 30.5% starch in this trial for feed. We take samples and 2 weeks and 8 weeks. And after 2 weeks of the feeding trial, the fish of the high starch level have a high level of liver size, and after 8 weeks, compared to the growth performance is reduced, but in the hepatic somatic index is no difference. And we found very interesting, the fish disease in liver is only observed in the samples of 2 weeks, and during the 2 weeks feeding, we found the cholestasis, we found the postprandial glucose with higher glucose, and we found that some immunity response in this group, in the high starch group, and in these 2 weeks phenotypes, we observed the nuclear dense and the fibrosis phenotypes, but when we feed to 8 weeks, there's always showed the very normal phenotypes. So we analysis this phenotype is when the 2 weeks hepatic glycogen is accumulated and the lipid is accumulated, and even the TBA and in the hepatic is very significantly increased. And this paper has been published in this year in the British Journal of Nutrition, and we confirmed the restoration of metabolic function related to inflammation, bile acid synthesis, and energy metabolism was related to the 8 weeks alleviation of the liver disease when we feed a high starch diet. And I gave a summary of large mouse bass. When we feed a high starch diet, the reduced growth performance was observed, and it will lead to the vascularization and fibrosis of liver tissue in the 2 weeks, but and lead to the metabolic disorder and acute inflammation response in 2 weeks, but not for 8 weeks. And the downregulate bile acid synthesis gene expression, and the adaptive response of liver injury caused by high starch diet is related to the bile acid metabolism. So how we use the bile acid to have an intervention to the fish. So for the cholestasis is a kind of disease, liver disease related to cholesterol metabolism. In the clinical, bile acid will be the first choice for the human to have a clinical result for the cholestasis, and other nutrients like methionine, taurine, glycine, and the cholesterol and vitamins, minerals, phosphorus will be participate in the bile acid metabolism, and some toxins will be negative, will damage the bile acid metabolism pathways. And there are two famous studies in this field for the HDC from the at a high colic acid species will be related to the glucose metabolism. There have several studies for the exogenous bile acid intervention in the fish diet, like including rainbow trout, yellow croaker, and also for the largemouth bass. And the diet bile acid could benefit the growth, lipid metabolism, and the gut microbiota. And in our another trial in the LachanceGroup efficiency and tolerance study for the registration in the MORA to the government, and in this study for the common carp, because they have a very low requirement for the lipid, and we found only 26 to 29.8% of bile acid will have a positive response to the growth performance. And for the histology, we gave the higher level of recommendation is 60 ppm, and we have a 10 time fold for the safety margin in both of group. We found in the even for the 600 ppm, we found very significantly cured the intestine structure for the high plant diet for the common carp. So we found the inflammatory response is decreased with the bile acid inclusion, and also for the liver structure in the 10 times for feeding, the office gave the very positive response of the health, and also for the inflammation response is decreased, and the lipid. We found the very significant increase the LDCA ratio to the TCA total cholesterol is increased. That means in liver, the LDCA will have the function to transfer the free fatty acid out of the liver. So this could be the one reason to clear the free fatty acid in the liver tissues. And also for the cholesterol, cholestasis biomarker, the plasma TBA is significantly decreased. And even we fed the fish with the HA and HDCA of long-term groups, and we found the common cancer synthesis still have a high expression of bile acid synthesis. And for the synthesis of fatty acid is decreased, and the lipolysis is increased. This is also the reason for the elevation of the fatty liver for common carp. And we determined the bile acid pools, the very important phenotype, although the glycine conjunctive bile acid is not the major bile acid in the pools, but we do find the significantly increase the glycine bile acid when we find the fish, and this could be potentially have some functions to increase the proportion to have relieve the common carp liver disease. So the function of glycine conjunctive bile acid will be further studied in this space or other spaces. This is the summary for the common carp. When we feed the high level of plant diet, the bile acid will have to modify the pools of bile acid and regulate lipid, regulate glucose, regulate and anti-inflammatory response, and to have the elevation response to the fish. And the second trial, we also for the registration in the garment and for the largemouth bass, and we give the response for this fish have a much higher level requirement for the bile acid. It's to the 475, but finally we gave the highest recommendation level is 300. in the garment, and we gave the fish with 300 milligram per kilogram in diet, and we found the elevation, they can be elevation, regulation of glucose metabolism by AKT-FOXO1 pathway, and gave the positive regulation to the lipid metabolism by the CAP-AMPK as REBP1 signal pathway. That's all of this will be increased the energy metabolism to have a positive response to liver disease. And finally, I will give some very shortly to have a crosstalk between the bile acid metabolism and the microbiota. So there have some negative response of the bile acid the intake. So in this paper, the bile acid bile salt from ox will be negatively will be give the very serious inflammation response to the Atlantic salmon, and also for the hydrophobic bile acid LCA also have a toxicity response to the animals. So in my lab, we found the fish amino acid pool, bile acid pool will closely relate to their nutrient requirement. For the carnivorous species like mouth bass, they have a higher level of requirement for the lipid, and in their bile acid pool have a much higher concentration of bile acid. This will be followed with the tilapia and the bullsnot bream. This species is kind of it's very it's kind of hybrid omnivorous. So we compared the several species for their bile acid pools. For the spotted and the largemouth bass, they have a very TCA is most most poor, and for tilapia is CA is the one the main bile acid pools. And this very interesting. It's bile acid pool is related to the response to the phenotype of the liver disease, and also for gut microbiota, for the human nutrition, and for the mammals is have been published in this year. There's a cohort for bile acid crosstalk research studies. So we also gave pertension in this in Sorrento, Italy, and for in our study of alternative for the condensated protein concentrate, we found that when we give the plant protein and yeast-based parabiotics, we have a crosstalk further crosstalk of microbiota to the bile acid pools. It's closely related. So in our lab, we will focus on the inflammatory and metabolism, and the study on the cluster to the bile acid pools. This will be our future study. So thank you very much for everybody. Thanks for listening. It's my presentation. Thank you very much.
Thank you so much, Professor Xue, for your insightful presentation about the replacement of fish meal and with the innovative plant protein such as cottonseed, single-cell protein. And now we have one question, Prof. Xue, from Dr. Ho. The question is, okay. The question is, one of the key points to replace fish meal for marine fish is EPA, DHA, ARA, fatty acid, but not seen in your evaluation. So,
I will give the answer for this question. For the marine fish, we focus on the amino acid balance, and we will give the fish oil to meet the requirement for the DHA requirement. The fatty acids have been balanced. So in our experience, when we give the enough fish oil, the fatty acids deficiency will be not a problem. But when we give the amino acid from crystallized amino acid, will be that we have some problems here for fish.
Okay, thank you. And another question, Professor. So, what are the possible reasons of for causing the metabolic liver disease, and the consequence of MLD, and how to prevent?
LLD what?
Metabolic liver disease.
Yeah.
So the question is, yes. You get the question? So
not yet.
What's the possible reasons for causing metabolic liver disease?
It's a quite systematically metabolic disorder. As I introduced in our trial in the spotted sea bass, when we give the high level of plant protein, the first problem is feed intake. But when we dissolve the feed intake problem, we can find the glucose, lipids, amino acid, all of them, the metabolism will be, it's not quite normal. So the most important phenotype is the cholesterol is not a deficiency problem, but the problem is when they synthesize enough cholesterol, but they cannot synthesize enough bile acid. Missing the bile acid deficiency will be the one important reason. So not for the plant protein replacement or for high glucose, high starch, all these phase, the phenotype is cholestasis. So the cholestasis will be dissolved by bile acid. So that could be one strategy to give the bile acid review. I think that will be very directly method.
Okay. So, thank you so much for your answer and sharing. I think this is definitely this will be very helpful for our future research related to replacement of fish meal with the plant protein. And if anybody audience, you have any questions you want to talk to Professor, I think you can direct contact her, or you put the QA box. So she will answer you after the presentation. So thanks again, Dr. Xue.
Okay, thank you very much. Bye-bye.
Bye. Okay, now we can move to our second speaker, who has double doctorate degree, doctor in veterinary medicine and doctor of philosophy in animal science. He is expert in salmon and trout production, nutrition and health, feed formulation, nutrition and health interaction. He has published more than 30 papers and he holds 2 patents. So I would like to call on Dr. Javier Gonzalez to present his topic on nutritional strategy to reduce feed costs. I think this is a very, very hot point. Okay, welcome, Dr. Javier. Now it's your time.
Okay, thank you very much. For the video, I think you have to give me, okay, there it is, I think. Yeah, okay, thank you. I will share my screen. Can you see my screen?
Yes.
Okay, very good. Well, thank you very much for the LachanceGroup for inviting me to talk to you and share some ideas on how we can, given the current scenario of high prices of feed ingredients, how we can reduce or try to reduce the cost of the feed, okay? So the overview of my talk, I will start first talking a little bit about feed ingredient prices and what are the reasons for this increase. And then we will discuss some strategies to reduce feed cost, including precision feeding and alternative feed ingredients. We'll talk about a little bit about formulation based on amino acids, and then also talk about some feed additives. Within the feed additives, we will spend a little bit more time on bile acids and show some scientific evidence, and then also some practical applications and conclusions. So starting with the feed ingredients and what are the main causes of increase? Well, I mean, I'm sure that most of the people listening to this webinar today know that the prices of the feed ingredients have increased since end of 2020. And there are several reasons that explain this increase. One of them is the supply and demand shocks. There is also a shortage of raw materials that have influenced the price. We have also seen prolonged supply chain disruptions, and also, of course, an increase in the freight costs. One other factor that have influenced is being the rising of fuel oil prices. We also have had some workforce shortages, especially related to the COVID. And of course, the consequence of all this is a higher production cost, not just for the feed producer, but of course also for the fish producer, okay, because of the excessive expenses in feed. So just to show a couple of figures, these are the price of corn. This is in dollars per bushel. If we see here the trend starting 2020, we can see a dramatic increase in the cost. The same thing we can see for soybean meal, also starting in 2020 with a significant increase in the cost. And unfortunately, the projection for this year and at least for next year is to stay in high prices. Maybe there will be a little decline, but the expectation is that the prices will keep up. And then if we see at the, let's look at fish meal. Fish meal has not changed as much as probably the plant proteins. And here we can see that in August 2020, the price was on average, this is for super prime fish meal, 1,400 approximately. And then if we look at the current price is around 1,800. Okay, so there is an increase, okay, over the let's say last couple of years, but it's not probably as dramatic as corn and soy. Nevertheless, if we look at the fish oil price, okay, and here we can see that since January in 2020 to current price, there has been a significant increase in the cost of fish oil. And of course, the consequence of this is that the cost of supplementing EPA and DHA in the feed becomes very relevant. Okay, so I mean, looking at this scenario, what we can do from a nutritional perspective, okay, and I will try to share with you some thoughts about this. Well, one of the first things is to think about precision feeding, okay? And here we talk about basically what are the nutrient levels in our feeds compared to the requirements. So the invitation here is to really review the requirements and be absolutely sure that we are feeding the fish with the right requirements and we are not overfeeding nutrients, okay? The other thing is to review the safety margins. Okay, normally when we analyze the feed ingredients over time, we come up with averages. Okay, for example, the average of crude protein, the average of digestible energy or fat or amino acids, and then we formulate based on those averages. But several nutritionists, what they do, including me, is to put some safety margins. That means that to the average, okay, nutrient level that we consider, we add a certain amount of based on the standard deviation of that average in order to let's give a safety margin. So let's review those safety margins and see if we are not probably overestimating those safety margins. The other important thing is to do an accurate and timely nutritional profile of the feed ingredients. Okay, so that's very relevant because when we have this such a high cost of feed ingredients, we need to be very sure that what we are using in our formula is what we have in the feed plant, right? So we have to exacerbate the analysis in terms of to have an accurate and reliable analysis of the feed ingredients for our formulas. And of course, the quality control of the feeds is also very important, because one thing is what we formulate, okay, in whatever we formulate and the nutritionist formulate in a feed factory is a theoretical formula, right? Because then we have to produce that feed, and then we have the final feed and the quality control. So what is the deviation between the final feed analysis and the formula, right? So this deviation is also important. So talking about this, what are the nutrient levels in our final feed? So when we analyze, and then we always, for our feeds, have a minimum limit and a maximum limit for a nutrient level. So are we looking at this kind of graph, okay, or are we looking at this kind of graph? Here, of course, we are oversupplying nutrients, okay, and this graph is where we want to be, where most of our analysis, okay, of the nutrient levels are within the limits that we have established. So this is another invitation to review the nutrient levels in our final feeds and see if we're not oversupplying of nutrients. So if we are oversupplying, when we have to do the necessary changes to go, of course, from graph A to graph B. This is where we want to be. Okay, what about the fatty acids? What about the highly unsaturated EPA and DHA? Of course, the minimum requirement vary according to species, and probably among the farm fishes, probably the salmon and trout are the ones that have the highest requirements of EPA and DHA. Nevertheless, we know that linolenic acid, okay, the C18, 3 and 3 can be converted by most fish into EPA. Okay, so there is, and this has been proven in many scientific publications, where it is well known that the fish contain these enzymes called the saturases and elongases that can convert linolenic acid into EPA and then into DHA. So the question is really, I mean, can we replace, or can I replace some of the EPA and DHA with linolenic acid? And I think that this is something that a few times is not looked at closely as it should be. And I think that we can certainly replace part of the EPA and DHA with either linseed oil, camelina oil, or canola oil. So we're not talking about here big amounts, of course, of EPA and DHA, but maybe some of it, you know, a certain percentage can be replaced with linolenic acid. Of course, there is other sources of EPA and DHA, like the macroalgae and genetically modified vegetable oils that today are commercialized. Nevertheless, probably the cost of these sources today are more expensive than fish oil. So the economics has to be done there, okay, what is more convenient. But definitely I think that at least one part of EPA and DHA could be replaced by linolenic acid. Alternative feed ingredients, and that's of course is another thing to explore. So can we reduce expensive feed ingredients in the feed, like especially fish meal or fish oil, or some other feed ingredients that are, you know, costly in our formula? So This is more a general comment to be alert on alternative feed ingredient prices and availability. So the purchasing people in the factory or in the feed company have to be, you know, very alert in terms of the prices and opportunities for alternative feed ingredients. And the other thing is to have good positioning of raw material. Okay, so if you can forward the contracts when possible, I mean, if you can find let's say convenient price for a feed ingredient, well, you can maybe forward these contracts for more time so that you can secure a lower cost. Nevertheless, when we talk about alternative feed ingredients, we have to always remember that the fish require nutrients and not ingredients. Okay, so we can replace expensive raw materials like fish meal, and today, you know, there are more than one example of zero fish meal diets that perform very well. So the important thing is to really know and have a good understanding of the nutrient requirements of the fish. In that same line of thinking, okay, we say, well, what about the crude protein? Okay, and how we should formulate, okay, on crude protein. And we know that most, not just the fish, but also land animals requires amino acids, okay, and not necessarily crude protein. So there is a general trend in the industry in order to formulate on amino acids and not on crude protein. Because when we formulate, okay, based on crude protein, and let's say this is just an example, right, of, let's say we need a minimum of 35% crude protein in the diet. Well, if we formulate on crude proteins and amino acids, but if we put a minimum on crude protein, we will also, we will always supply some non-essential nitrogen, okay, that the fish really don't need. And sometimes it even becomes an excess, and that nitrogen has to be metabolized. And when metabolized, it will save, I mean, it will expend energy as well. So let's say that if we only formulate on essential amino acids, our crude protein level will lower, and the feed cost will also go down. So, but to do this, we have to have a very good understanding of the essential amino acid requirements, but not also the requirement, but also the balance, the adequate balance. Some years ago, I would say at least 20 years ago or more, especially in the poultry industry and in poultry nutrition, people started talking about ideal protein. And what the ideal protein concept is basically the adequate balance of amino acids, essentially, I mean, related to the level of lysine in the diet. And this information is today used in feed formulation, not just for poultry, but also for swine. And this is a concept that in fish is starting to also be used. So what is the right balance? And that's something we need to understand really in order to apply this concept of formulating on digestible amino acids, but not on crude protein. Enzymes. This is another alternative to reduce feed cost. Of course, here we have to make a big distinction between cold and warm water species, okay? Because the enzymes, okay, the activity of the enzyme depends on the temperature. And here we have some examples like the use of phytases, carbohydrases like silanases or beta-manase, amylase, beta-glucanase, and some proteases, okay? But all these carbohydrates and some of the proteases, what they do is basically to improve the digestibility of carbohydrates or protein. And then on the phytase is basically what is a phytase? Phytase is an enzyme that cleaves the phosphorus, okay, in this inositol ring that is called the phytate complex of phytic acid. Okay, so most of the phosphorus in plant raw materials is in the form of phytic acid, and the phosphorus is not available for the fish. So what we do by adding phytase is just cleaving, okay, the phosphorus and some other trace minerals in order to make that phosphorus available for the fish. So what I said before is that the activity of the enzymes depend on the temperature, So, for example, in salmonids, we can grow salmon in 5 degrees Celsius up to, let's say, 14, 15 degrees. But in warm water species, of course, you go much higher in temperature. So this is something you really need to consider when thinking about using enzymes in the feed, and how this curve behaves might differ between different enzymes and different species. Okay, so this is something to think about as well, because here I put a red line that is a linear increase, but probably this increase in activity is not linear. Okay, maybe the activity decreases up to a certain temperature and then declines rapidly, or probably it will decline rapidly at the beginning and then it will decline more slowly. We don't know, but this is kind of information that we need to ask, of course, the suppliers of this type of enzymes. And then on the phytase, as I said before, on average, 70% of the phosphorus in the raw materials, in vegetable raw materials, is in the phytic form or a phytic acid form that is not available, and 30% it will be in the non-phytic acid form and is available. By using phytase, we liberate the available phosphorus. Normally, on average, we can say that by using phytase, we can liberate between 0.15 and 0.2% available phosphorus in the formula. And of course, these phytases are used at low inclusion levels between, let's say, 50 grams per ton up to 200 grams per ton, and they replace feed phosphates that are used in much higher volume. So you also open space there in the formula, what is also a saving. Okay, and of course, there is a benefit of reducing the phosphorus loss into the environment. And then we have some other feed additives like prebiotics or symbiotics. That is a combination of probiotics and prebiotics, postbiotics, etc. There is new names for this kind of products that in general they improve gut health and development, improve growth and FCR. And one important thing that Dr. Xue Min explained before is about how a higher inclusion of plant ingredients will increase the level of anti-nutritional factors or non-starch polysaccharides in our feeds. And these products could also be opportunities for increasing the level of plant proteins in our diets. And then we have also phytogenics to replace, some of them to replace synthetic nutrients. For example, today there is a natural source of choline to replace choline chloride. There is also polyherbals to replace vitamin C, vitamin E, and also methionine. And there is plenty of phytogenics with anti-inflammatory and antioxidant activity that can also aid in the replacement of expensive raw materials like fish meal with plant proteins. And then I will just talk about this feed additive. The name is guanidinoacetic acid. There is very little experience in fish on this product, but I just wanted to mention it because I found it very interesting. Guanidinoacetic acid is used in the poultry and in the swine industry in order to reduce the energy in the diet. Okay, so basically, what is guanidinoacetic acid? Is synthesized in the kidney from arginine and glycine. And this guanidinoacetic acid is converted in the liver into creatine. And then the creatine goes to the muscle, and creatine is phosphorylated into creatine phosphate. But when doing that, it converts ATP into ADP. So basically what creatine does is it works in recycling of energy in the muscle. So basically makes the energy metabolism in the muscle much more efficient. Okay, and that's how it allows to reduce the energy level in the diet in order to obtain the same performance. So this is another way to reduce cost. I don't know what will be the effect of this compound in fish, because as I said before, there is very little experience, but I think it's a compound that will be interesting to look at. And then we come to the emulsifiers to improve fat digestibility. Here we have the lysolecithins and the polyethynyl glycol resonylate. As you know, fats are mainly composed of triglycerides, and although fats are not water soluble, in fish especially, the digestion takes place in an aqueous environment. in the intestinal tract, where it is hydrolyzed by lipase into fatty acids and mono and diglycerides. So the mode of action of the emulsifier is to increase the active surface of fats, allowing the action of lipases, and to favor the formation of micelles consisting of lipolysis products. And the micelle formation is an essential step for lipid absorption, as it creates a diffusion gradient and promotes absorption. So within the emulsifiers, of course, the bile acids are a very interesting opportunity, an alternative. So what are bile acids? Bile acid participates in the emulsification of fats in the intestine, transforming large fat droplets like this one here, okay, into smaller droplets, okay. So they, what they do, the bile acids, is to promote and improve the action of the lipase enzyme to hydrolyze the fat in the intestine. So that's one of the functions of bile acid, but bile acids also, and this is very different from the other emulsifiers, is that bile acids also, what they do is to, they activate the pancreatic lipase, okay, so that lipase can do its work in hydrolyzing the fats. And also the bile acids, they also are important for the absorption of the long chain fatty acids. Okay, the short chain fatty acids normally are absorbed without a carrier, but in the case of the long chain fatty acid, they do require a carrier to be absorbed, and then the bile acids also participate there. So bile acids really participate in all the steps of the fat digestibility. When we talk about the importance of bile acid, there is an inverse relationship between the fat digestibility and the fecal bile acid loss. So normally when we see a decline in the fatty acid digestibility, we also can see an increase in the loss of bile acids into the feces, okay? And this was observed in rainbow trout. So what about bile acid and non-starch polysaccharides? Okay, and this is also a study that was conducted in trouts. This study used 4 treatments. So we had a low non-starch polysaccharides, for example, 1.2% without bile acids, and then low non-starch polysaccharides with bile acids, and then a diet with high non-starch polysaccharides without bile acids and high non-starch polysaccharides with bile acids. So in this experiment, what was observed, if we look at the digestibility of the fat, we see that the inclusion of non-starch polysaccharides, comparing this column with this one here, you can see that there is a significant reduction in the digestibility of the fat. Nevertheless, when we include bile acids on this diet, the digestibility increases significantly. Okay, so there is an interaction between the non-starch polysaccharides and the bile acids in the diet. So again, in this graph, this is the same trial. So we have the low non-starch polysaccharides plus bile acids and the high non-starch and then plus bile acid, and we look at the fecal loss of bile acids. We see that in the adding bile acids to a low non-starch polysaccharide diet increases the bile acid loss because there is no need for more bile acids. Nevertheless, when we include a high level of non-starch polysaccharides and we add bile acids, we see a reduction in the losses of fecal bile acids and also of the cholesterol in the feces. So this is another trial, but this is now in tilapia, okay? And they were looking at different levels of bile acids inclusion in the diet. This trial was for 9 weeks, and we see that the levels were from 50 to 1,350 grams per ton of bile acids. Basically, the level of 150 was the one that performed the best in terms of weight gain, feed efficiency, and hepatosomatic index, as well as the level of AST enzymes, liver enzyme, okay, in the blood. Another trial in trouts, also with different levels of bile acid inclusion from 300 to 1,200 grams per ton of feed. And again, here, the best growth was obtained with 300 grams per ton and 1,000 grams per ton of bile acids. And all the levels of bile acids improved the FCR, okay, in the trial. This study was made in sea bass, and again, different levels of inclusion of bile acids from 80 grams per ton to 600 grams per ton. And this trial was also shared with you with Dr. Xue Min in the last in the previous talk. So I will not go into much detail here, but basically the SGR or the growth was improved with 240 to 300 grams. The pathosomatic index was reduced with the highest levels of bile acids. The liver fat was also reduced with intermediate levels of bile acids, and the ALT enzyme in the blood was also reduced with the use of bile acids, indicating that the use of bile acid will improve the liver health. And this graph was also shown before, but basically in this study they concluded that the level of 475 grams per ton of bile acid was the best in terms of the growth performance of the fish. Well, regarding soybean meal now, we know that high levels of soybean meal, over 20%, induce enteritis in salmonids, and thus it's been very, very well studied. And basically there is a pro-inflammatory response and a loss of the tight junctions in the intestine. The soybean meal also decreases the villi length, increases the villi width, and induces mucosal fold function and inflammation. And also soybean meal induces atrophied hepatocytes, reduce the ability to digest and absorb lipids, okay? And also impairs the bile physiology. So the use of high levels of soybean meal have been related to several gene responses. I will not go in detail here because Dr. Xue Min went very much in detail with a very good talk about how plant proteins can influence the bile acid metabolism. But basically, there is a reduction in the conversion of cholesterol into bile acids by reducing the expression of the CYP enzyme. And also there is a disruption in the absorption of bile acids in the intestine for the enterohepatic recirculation of bile acids. So there is basically an alteration in the bile acid metabolism with the use of soybean meal. So this is a recent study that was conducted in order to look at the effect of high levels of soybean meal in trout diets with or without bile acids. And you can see that they use 5 treatments in this experiment. They have a control diet with a high level of fish meal, 25%, and then 2 levels of soybean meal, 30 and 40% with and without bile acids. All the treatment from 2 to 5 contain 10% fish meal. And you can see that looking at the histological scoring, okay, in the intestine, that when adding bile acids, both to 30% soya and 40% soya, decreased the, let's say, the alterations in the histology. And this was basically the scoring based on absorbing vacuoles, lobed cells, inflammatory cells, lamina propria, and mucosal fold fusion. Okay, so different alterations of the intestine. And then looking at the liver, in the low level of soybean meal, there was no effect, significant effect of the bile acid. Nevertheless, when they increased the level to 40% and including bile acid, there was also better histological analysis or scoring in the liver. And there's some other studies that have observed similar effect of the bile acids in trouts with high levels of soybean meal. Okay, so this is not the only study showing this beneficial effect. And we know that basically bile acids, what they do also is that this is very interesting because this is taurocholic acid, binds to some receptor in the epithelial cells in the intestine. And one of them is the TGR5 receptor. and by activating this receptor, there is a reduction in the tumor necrosis factor alpha, and also an improvement in the tight junction in the cells of the intestine, and also through this other receptor, the ASBT, there is a cascade of reactions at the nuclear level in order to reduce the production of pro-inflammatory cytokines, and also reduction of the nuclear factor kappa beta that is involved in the inflammatory response. So there is high evidence of this, indicating that bile acid will reduce inflammatory effect. Okay, so a practical example. So how can bile acids look in not just from the health perspective on the liver health, but just on the digestibility perspective. If we have that bile acid can improve, and this is just an example, okay, because the increase in digestibility will depend on the species, the diet formulation, the fat level in the diet, etc. But this is just an example, okay. If we, let's say that we can improve in 1.5% the digestibility of the fat, and we have a diet which has a gross energy of the energy of the fat is 39.8 megajoules per kilo. The content of fat in the diet will be 20%, and we have the fat digestibility without bile acid will be 90%, and the fat digestibility with bile acid will be 91.5%. So basically, without bile acids, the energy level in this diet, okay, supply by the fat, okay, will be 7.16 megajoules. If we add bile acids and then we increase to in 1.5% of the digestibility, we come up with 7.28 megajoules. So we have a difference of plus 0.12 megajoules per kilo feed by adding bile acids. So what are the possible applications? Well, I can formulate with less 0.12 megajoules per kilo feed and expect the same performance of the fish, or I can formulate with the same level of digestible energy and expect an improvement in performance. So those are the two objectives. But if I want to, if my objective is to reduce the feed cost, probably I will reduce the level of energy in the diet, add the bile acids, and then expect to have better cost per kilo of fish produced. Other potential benefits of bile acid inclusion could be improved EPA and DHA retention. This has not been tested as far as I know. Nevertheless, if we consider the bile acids can improve the fat digestibility, maybe we can improve the EPA and DHA retention, and of course that will be another way to reduce the cost, but because maybe we can reduce the level of EPA and DHA a little bit in the diet, that is very expensive. Also increase pigment absorption, and this can also, if we can improve the pigment absorption, then we can maybe reduce the level of pigment a little bit in the diet to also save some cost. And then a higher utilization of fat-soluble vitamins. So this effect can also reduce the feed cost in the diet. So to conclude, there is a series of nutritional tools to reduce feed cost in fish feeds. The use of bile acids is among the ones that to be seriously considered. Each of these alternatives must be evaluated under the market situation of each region, country, and company. The recommendations indicated in this presentation are based basically on my experience. Of course, this is important to communicate. And this is very, very important that it is recommended that each company organizes a team to address all possible tasks and make an action plan to reduce feed costs. And that includes that we need a team with the nutritionists, the purchasing people, the quality control, the supplier, etc. We need to have a team to address the high cost of feed ingredients and how we as a feed company can reduce the feed cost. Okay, with that, thank you very much for your attention, and I will try to answer any of your questions.
Thanks, Dr. Javier. Thanks for your sharing on the reducing cost for the aquaculture. I think now we have 3 questions in Q&A box. Okay, the first is, what's a feasible way to measure digestible amino acid? Dr. Javier?
Sorry, what was the question again?
What is a feasible way to measure digestible amino acid? Okay, this is from ROMI.
Yeah.
Yes.
Yeah. Okay, I understand the question. Yeah.
Yes.
Well, it's very feasible. We have done this in salmon and trout for many years, you can. I mean, the most used is in vivo digestibility because it's the most reliable. I mean, I will not discuss the technique now, but if someone wants to have more information, I can send you the information on how we do this in salmon. But basically is to feed the fish with the two different formulations, one with a high concentration of the feed ingredient that you want to test. You use a marker, and then you can do some stripping, or you can collect total feces, but this is absolutely doable. And you can calculate the digestible amino acids in the different feed ingredients. Yeah.
Okay, the second, I think, from Dr. Ho. It's very long question. It is interesting to approach of reducing cost by increasing nutrient in more bioavailability and using additive to enhance nutritional approach. To enhance nutrient absorption and digestion. However, feed formulators will face a common question that increase formula cost once adding additive before they wait for animal performance to be payback. So the question is, what's your recommendation to feed formulators?
Yeah, okay. Well, that's a good question. But you're absolutely right. That sometimes we believe that by adding additives, okay, we will increase the cost of the feed and not reduce the cost of the feed. So I think that there is not a clear answer to that question because it depends on the reality, okay, of each company. Based on their formulations, but based on the price of the feed additives. I think the most important thing is to really do good testing. Okay, so let's say that the suppliers will provide you with very good research, reliable research, or you, in your own experience with your own fish, you do your own testing. Okay. But this is, of course, depends on the effects. What we see, for example, I mean, my view of this in general terms is that, and this is being not explored in depth, okay, and it relates on how we can replace expensive feed ingredients with less expensive feed ingredients, and how the additives can help us to reduce the cost. So, of course, the reduction in expensive feed ingredient has to be, let's say, must cost more than the feed additives. If not, it doesn't make any sense.
Okay, Dr. Javier, I think we have quite a lot of questions from audience. Now we have next question. Let us answer one by one. So could you introduce some information on salmon farming in Latin America, the main diseases and the bottleneck, since we know the Latin America is the main farming area for the salmon. So please.
Yeah. Well, the salmon production is only in Chile, okay, in South America, and there is some trout production in Peru, also in Colombia. But the main volume.
Please mute your microphone.
Okay, so the main volume is in Chile. Chile this year will produce approximately close to 850 to 900,000 tons of salmon and trout. And the main diseases, I would say, is one of the main problems is sea lice, okay? Sea lice infestation. And this is not only in Chile, but also in the other countries that produce salmon. And I say that is a big problem because sea lice induces immunosuppression, and is also the open door for many diseases. And we also have some bacterial diseases that are a big problem. One of them is Rickettsia salmonis. Okay, that is the #1 problem. We also have some BKD, bacterial kidney diseases, yeah, I would say those are probably the two main bacterial diseases. And there's some other challenges related to gill disease, what we call the gill complex disease, that is a mix of virus, bacteria, environmental toxins, amoeba, that is a parasite, etc. So I will, if you ask me to summarize what are the main issues, I would say it's sea lice, Rickettsia salmonis, BKD, and gill disease. Yeah.
Okay, thanks, Dr. Javier. So just now you mentioned the sea lice. So any solutions or strategy can prevent or treating sea lice so far?
Yeah, that's
anyway.
Yeah. Well, I mean, sea lice is today is mainly treated with antiparasites. Okay. And then in the northern hemisphere, especially Norway, they are trying other methods that are not therapeutic, okay, that are mechanical treatments, either with fresh water, with the temperature, with the high speed water current, etc. So there are different mechanisms or system to control sea lice, but the most used are antiparasites, okay, so medicine, and these mechanical treatments. And of course, everyone is investigating either a vaccine, people are investigating about phytogenics and how phytogenics can, reduce the lice infestation, immunomodulators, etc., etc. So there's some other, there is several people working on different alternatives to control sea lice, because sea lice for the salmon industry has a cost of more than 1 billion euros per year. So it's a huge amount of money and losses. Yeah.
Yes. Okay, thank you. So one more question for the shrimp feed. Nitrogen utilized less than 50% of shrimp body. Rest go to the water and cause pollution. Okay, I think this is from audience. Okay, so when we feed to the shrimp, we waste the feed, but the utilized rate is only less than 50%. That means maybe 30 to 40% for growth. So the rest are discharged into the water. So like ammonia, like a fecal method. So the question is how to learn from experts. What's the key bottleneck for that? Could the efficiency to increase to 80% or higher? How to realize that? I think this question a bit confused. Let me understand. Okay, so I think the main point is when we feed to the shrimp, the feed 1 kilo, maybe only 30% for growth, and the rest discharge in the water. Okay, so the question is how to improve the rest 70% or 60% for the growth. You get my point?
I mean, if I don't know if I understood,
maybe
but
increase the
utilization
rate.
I think the question is related to the loss of undigested feed into the environment.
Yes, correct.
Okay.
Yes.
Okay. So if that is a question, then of course the loss is linearly related to the FCR, to the feed conversion rate. Okay. So how we can improve the feed conversion rate. Okay, and that's basically the main question, because the better we have a feed conversion rate, of course, the less will be the losses into the environment. Yes. And that includes nitrogen, phosphorus, everything. So, just an experience from my side. few years ago, the feed conversion rate in the salmon industry in Chile was 1.4, 1.5. Okay, and today, okay, the average is 1.1. Okay, and maybe a little bit more than that. So what have we done really in those years? In my opinion, basically trying to improve the health situation, reduce the mortality, and improve the knowledge of nutrition in salmon, so that we can improve the FCR. And that is being basically increasing the fat energy in the diet. So the fat energy levels, I mean, the fat content in the diet has increased significantly. And that has implied a reduction in the FCR. But it's several measures that have been taken. It's not just nutrition, it's management, biosecurity, it's a mix of things. But basically, the idea is to reduce the FCR as much as possible, and of course reduce the uneaten feed. Okay, and that's also a big control on the waste feed. So there is a lot of technology that has been included in how we feed the fish, so that we waste the least possible feed into the environment. I cannot hear you. I think you're in mute, Dr.
Okay, sorry. Thank you. Thanks for your answer. I think that as a rest of question, I can try to answer. What's the source of bile acid? I think so far all the bile acid extract from the animals, but after that, the process, chemical synthesis is very, very complicated. I think LachanceGroup has a lot of patent on this processing. You can leave the message, Suguma. You can leave the message. You can talk to the technical department from LachanceGroup. Okay. And for the, let me see, the rest. Next talk. My question to the speakers. Okay, this question from Hakim. What are the different ways to estimate the extract FCR in a wet lab feeding experiment? Doctor, you get my point? So the question is,
what is the difference?
Well, what are the different ways to estimate the exactly FCR in wet lab feeding experiment?
Yeah. Okay. Of course, in the wet lab is much more, let's say, reliable, because you can collect the uneaten feed, right? So you can, and also in the lab you can weight the dead fish. Okay, so in a wet lab it's very it's more accurate. Okay, the FCR, because you are, as I said before, you're collecting the non-eaten feed or the uneaten feed. Okay, and you can weigh the feed, and so you know exactly how much feed the fish consumed, and then you can have also a good control of the weight gain, so you can have a very good estimate of the FCR. And if you have dead fish, you can weigh the dead fish and include that dead fish into the weight gain. Okay, but in farming conditions, of course, you are not measuring the feed that the fish doesn't eat, right? So basically you have what we call an economical FCR. That is basically how much the fish gain versus at the end of the cycle, right? So is the fish you harvest, okay, and you kill at the end of the cycle, of the production cycle, versus the feed that you gave during the cycle. So this is what we call the economical FCR. Yeah, so certainly the lab, the wet lab FCR is much more accurate.
Okay, thanks, Dr. Javier. I will answer the next question. This question from Ayanan. Ayanan, may I know your location? Because you know, bile acid has been exported to 45 countries all over the world, from Latin America, Middle East, and Asia, of course, Asia. So you leave your message. I think in your country, definitely they have the dealers. and our LachanceGroup technical department or sales management, they will contact you. Okay, I think if I'm not wrong, you can search online. There are plenty of papers on doing the research on bile acid. Okay, let's move to Dr. Javier, one more question from Alan Ho. What interaction do you see between GMO and the nutrition in the strategy to reduce cost?
Well, the GMO at the end is based on market. Yeah. It's a market demand, right? So for example, I mean, if the market where you are selling your fish, okay, requires non-GMO, well, you cannot use it. So it's really about a market requirement. So where you don't have restrictions about using GMO, like soybean meal or whatever other GMO feed ingredient, well, why not to use it? If it is cheaper and allows you to save some money, well, I don't see the reason why not to use it. But at the end, it's a market-driven decision. Mm-hmm.
I have one more question. You just told the high price of feed, but what about the price of fish? Price increase, which give farmer the benefit at the end.
Yeah.
From Mubarak.
Yeah, but I see the point. The problem is that the price of the fish is not always going up. And so sometimes, the feed prices the feed ingredient prices go up, but not necessarily the price of the fish goes up. There is no linear relationship between the two. There are several examples where we have experienced high prices of feed or feed ingredients and a low cost of the fish, because at the end the fish, the cost of the fish or the price of the fish is also much more related to let's say offer and demand. So yeah, if you have, of course, a higher fish price and higher feed cost, and they are at the same time, okay, good news, but it's not always the case.
Okay, one more question, Dr. Javier. So this question from Asra Ali. So his question, can we reuse the water having fish fecals and undigestible feed? I think this is a water management or system management question.
Okay, can we reuse water having
fish fecal? You see the question?
I don't know if I understand the question.
Yeah, I try to understand.
Is that related to a recirculation system?
Yeah, I think the water, when we Ali, I try to understand your question. Your question should be, so once you use this water, after discharge, you want to reuse again. As Dr. Javier just mentioned, recircle system. Okay, actually this water we can use, but remember the water quality, especially for the ammonia nitrogen and ions ammonia nitrogen. You must treat properly. Okay, because as we know, if pH go up, your ammonia toxic from ammonia nitrogen it's increased a few times, 10 times or 20 times. Okay, 4 times increase. So remember, remove. Another, because the different fish, they have the different requirement for the environment. Okay, if the water too cloudy, maybe cause some problem, like the parasite, like the harmful bacteria. If the, let's say the fish under stress, if your water quality become worse, so may your fish get problem. Okay, of course, this is very complicated. for the water chemistry. You can leave the message. I think after the webinar, we can talk about details. Okay, from the mud pump, from the intensive farming, from the rush system, how to treat the water. Okay, that is the standard.
Yeah, I can just very short. I can just comment this.
Yeah,
that,
please go ahead. Yeah.
Yeah, that at least here in Chile, we are using, of course, the recirculation system are becoming very popular. More than 50% of the smolts, of the small fish are produced in this recirculation system. We reuse 95 to 98% of the water. That means that, and we use, of course, high technology to remove the undigested feed and the feces. We use biofilters that do this work, and we also use disinfection system like ultraviolet light in the water in order to control the pathogens. Okay, so yeah.
Yes, yes. Okay, I think we have one more question, Dr. Javier. This is to you. Nilo, this is from Nilo. The most difficult part to justify in the formulation is the use of enzyme and additive to improve fish diet performance. How can we convince the management to utilize bile acids? Okay, and utilize more plant-based protein, and besides field trial, which require longer time to justify. I think in your presentation you have such kinds of the trials. I think you can share more about this, please.
Yeah, well, basically, I think you have to go into the economics, right? Because the manager, the general manager, will always look at the numbers, okay? And so basically you have to say, okay, and do the calculations, no? And of course, the feed formulation software is probably the best tool to do that, okay? So, I mean, just you need to have accurate information on the benefits of the additive. So when you have good information about an additive, for example, an enzyme or whatever that will improve the digestibility of that nutrient, or I don't know, it will allow you to reduce the energy in the diet, or it will allow you to lower the phosphorus level in the diet, or whatever. Okay, all that has to be accounted economically in the feed formulation. So the thing is that you have your standard formula with this cost, and then you can develop different formulations with the different feed additives, okay, and with the expected improvements, and then compare, you know, and then if you are saving money, well, you know, that's what you need to show to your manager, okay. And of course, you need to consider in this economic evaluation the performance of the fish, because some changes that you do in your formulation will impact the performance of the fish, either improve or reduce a little bit. But at the end, your customer, the fish producer, okay, is the one that will have to evaluate the cost of the feed versus the performance of the fish, and then calculate the cost per kilo of fish produced, the cost of the feed of kilo of fish produced, and that's the main outcome of this. Yeah.
Yes, I fully agree with Dr. Javier, because I'm from the aquaculture industry, for the farming side, for the system side. So the farmer, the need from the farmer is high yield, high production, high survival rate, healthy fish. Okay, when you add some additive in your feed, I mean for the feed mill company, when you add some additive, the function should be improve the quality of the feed. Of course, at the meantime, Dr. Javier also mentioned the economic performance. It's also very important. I think there is a balance. Okay, if you want to have further discussion, I think you also can talk to Dr. Prof. Xue Min, because she also did a lot of research on this part in the past 12 years based on her presentation. Okay, I think if you have any other questions, you just leave to Q&A box. Then we will try to entertain this question in the afternoon. Oh, I think now we move to, first I'd like to thank Dr. Javier, share too many useful information. Definitely we'll learn a lot, okay? And also thanks for your patient answer our questions. Okay, so I think now we move to our tea break, short break. From 10:30, we come back. 10:30, we come back. So later, Mr. Akter from Bangladesh and Dr. ROMI from Indonesia, Jakarta University will share more information. Okay, so now we move to tea break. So ladies and gentlemen, welcome back to our webinar. Now we are moving to our third speaker, who is Dr. Eroldoğan-Zhang Wei-The from Bangladesh. He is a master's in poultry science from Bangladesh Agriculture University and MBA in Dhaka International University of Bangladesh. He is animal nutrition consultant. He has been working in the poultry and aquaculture industry for 20 years and serving for SB poultry, fish and poultry feed, fatty head, fish and poultry feed, Western feed, and so on. He is an expert in feed formulation too, and conducting technical training for officers from ACI, a very big, very huge company, Farmer Ross. Engineer, and please welcome Mr. Akter to share the topic of current status of fish production and the market trends in Bangladesh. Mr. Akter, please.
Okay. Yes. Thank you. Can you hear me?
Yes, I can see, but not so clear.
Not so clear because I'm not so. I think there may be Wi-Fi capacity.
Okay.
Thank you I'm very happy to have a chance to be associated with you all, especially on the webinar of change and challenges in aqua nutrition. So I am Akter Jaman already introduced with you all by our moderator. So I have been working especially for Aquafeed and poultry feed in Bangladesh since 20 years. So I am So today's topics, especially for me, current status of fish production and market trends in Bangladesh. Also, Bangladesh is overpopulated country in Southern Asia. So due to overpopulation, we have to focus on supplying quality protein, especially for animal protein. So we have to be concentrate on production of fish here in Bangladesh as a protein source. And so from our Department of Fisheries of the government of Bangladesh, trying a lot of especially some incentive program and the foreign investment welcome program and the extension services for feed and fish fry. So Bangladesh is getting along with good progress, especially in fish production, and they are trying the level best to be one of the most important countries for inland water fish production. Already they have achieved that.
Like that.
Yes,
sorry
please.
for disturb you. So large your screen. So you clear? So please enlarge your screen. No, not this way. You click the right corner.
Is it not?
Okay, so you click the broadcast, the button of broadcast, not this red corner below and the red corner.
Is it not clear now?
Not clear. You 你给他发个信息。So I think. So you know that. Yes, that one. Right. Yes, you clear this button.
Is it?
Yes,
Is
that's
it clear
correct.
now? Is it clear now?
Yes. Now
Yeah.
very clear.
Okay.
Please
Thank
go
you.
ahead.
Thank you for being it lit. There is a picture actually to see how actually a lot of species of fishes, and they are culturing actually in the pond mixed culture, and our national fish is Hilsha. It's contributing 1% to GDP, and you know, in dry season there is a funny harvesting of all the farmers. It's a fun to be our traditional system of harvesting fish. So we are actually practicing fish culture from the traditional practice, but it is now getting improved by the intervention of foreign companies and as well as local companies through feed production and quality fish seed, fish fry. So fish and fisheries sector contributes 3.61 to Bangladesh national GDP, and it's a little bit increase, and sometimes it's within 3.5 or 3.6 and around 24.41. Last year it increased a bit around 25 to the agriculture GDP. And last 10 years on average growth performance of the sector almost 9.1, but 2 years ago it was 8.19, but a little bit increase. So we are getting progress within this percentage. It shows that we are getting increase. The global position of rank of fish production in Bangladesh in inland water body is we are in 3rd position, and fish production in close water body we got position of 5th, and overall including marine we have got 11th position. So it indicates that for inland pond water where quality feed and quality fish fries and technology pond management are the main keys to come to the position of 3rd. So it is the good sign of getting progress, especially in feed production. And you know, water land is very limited. Only inland 47.6 lakh hectare, and inland capture occupies 39.27, and just a seasonal culture area is on an average of 8.33. So much limited, you know, but open area is especially river and marine area. That's a bit good for us, but we don't have enough technology to increase our marine fisheries. But we are very centric to inland capture pond culture for the fish production. But in the coastal area, there are some practices, especially for free shrimp and prawn, but that also limited, Bangladesh possesses a large inland water that's comprised, rivers and open large water bodies for diversified fish resources. 260 freshwater species are available here in Bangladesh, and 475 marine fish species in the country. It is good sign to get progress for feed production, but we need technology and foreign investment for getting it increased. Fish production in Bangladesh has increased for the last 2 decades, and it indicates that from 17.81 lakh metric ton, it was 2001, and just 2020, it was 46. So good sign of increasing of fish production through the technology and other quality feeds and inclusion of feed additives. The production depends on the pond culture and technologies and other functional feed additives, and especially for extension works from government and private sectors. Now private sectors are booming up to facilitate the fish production in Bangladesh. Here I would like to mention some important feed mills. Even though they are very focused on poultry feed production, now they have concentrated their fish feed production, and especially for floating fish feed, they have invested a lot. Just Nourish, it's a big company, especially for poultry, fish and cattle. Then Paragon, Kaji, Akter, Quality, Spectra Hexa. Spectra Hexa has joint venture company. They have a joint venture with Taiwan company and one of Thailand company. And RRP is a big investment oriented company, especially for fish feed production. And Nahar, very recent, they have set up big fish feed production unit. They are getting progress. So these are the company who are actually focusing now on for floating fish feed, even though previously they practiced with sinking fish feed. This is a graph, and you can look at the graph that it is increasing. Every year it is increasing. It's a good sign for getting progress in feed production towards fish production through quality feed and quality fish fry. And the last 5 years, that is the figure from 41.3 lakh ton to 46.21, a good sign for increasing. And why I have given it here to be centric to the parameter set by our government. As a nutritionist, I have to follow this parameter, especially for carp fish feed, nursery, starter, grower, and finisher. We have to follow protein percentage. That is very important. 28% CP to 21% CP, nursery to finisher. We have to follow this parameter. This is very important. And fat percentage from 7 to 4 on the basis of ages of fish, especially for carp feeds. So this is the set standard from our government part. As a nutritionist, we have to follow this standard to get the quality of our produced feed. And same way for catfish from CP 32 to 24, from nursery to finisher, and fat from 8 to 5. This is the way, and the other parameters also here mentioned. And the same way for koi fish, same way for tilapia. We have progress very dramatically to increase tilapia fish across the country. It is being cultured, and there is a good standard of CP for tilapia fish feed from 30 to 24, from nursery to finisher, and the fat 6 to 8. Same way for prawn from CP 32 to 28 and fat 7 to 5, from nursery to finisher. This for prawn. And shrimp, it is saltwater shrimp in coastal area of Bangladesh that shrimp is being practiced, and it has got some export market also. So it is getting a contribution 1.39 to the GDP, a good sign for us for increasing export market, and government has set up the parameter for shrimp feed, for producing shrimp feed. CP will be 40 for nursery and 32 for finisher. In between, we have to follow the CP percentage and fat from 5 to 4. So this is the fat percentage from nursery to finisher. So why I have given you the idea of starch source, you know, starch is very complex. If we consider protein from a plant source, so we have to consider how complex it is, because you know non-starch polysaccharide is the main barrier, because fish has some species to species shorter bowels, so it's very difficult to digest complex starch. You know, starch is very important carbohydrate. If there is any stress, that time starch, I mean glucose, is very important for minimizing starch. So utilization of glucose from starch, how it will be broken down, you know, in shrimp there is no liver and pancreas, but a little bit participation of the liver and pancreas, that is hepatopancrease activities. So we have to consider in what way complex starch will be digested and the ingredient will be selected. That is very important from nutritional point of view. And excess carbohydrate, you know, all the times in feed causes some intestinal problem in Aquafeed. The reason is that gut length differ from species to species. That's very important area we have to look into this matter, how the plant source protein item will be selected. Then we are going for a protein in the same way. Protein is very complex part, you know, our hair also protein, it is a structural protein, bone also protein, it is a structural protein, but it's not so easy to digest. So we have to consider easily digestible protein. That is very important source is animal protein source. But nowadays animal protein source price and other plant protein source price has gone up. So we have to consider in what way there will be mix of plant protein source and animal protein source. But even though we know that animal protein source digestibility is good, amino acid profile balance is good, and there is less anti-nutritional factor. In the plant protein source, there are some anti-nutritional factors, especially we can mention in the cottonseed, gossypol, and some anti-nutritional factors in soybean meal that have to destroy it by heat. Otherwise, it will not be easily digested, and it will hamper also other nutrients. So it will get also imbalance of intestinal health. So that is very important. We have to consider animal protein. Animal protein doesn't contain any anti-nutritional factor. That's very important issue. And attractiveness of animal protein is very important for fishes. And very rich and comprehensive nutrition. Always we consider that all the essential amino acids will be available in the animal protein source. And from digestible point of view and the absorption and utilization of protein, especially for amino acid, is easily digestible if it is from animal protein source. That's why we have to consider it as a nutritionist. We have to select how much animal protein source will be added to get it balanced in amino acid profile. That is very important to get a good FCR. This is the point, you know. Even though we have to consider crude fat, and for floating fish feed, we have to consider 16 to 20 kgs of oil. So that's why if we put oil, how, or if there is any crude fat, how it will be digested. Emulsification is very important. So what is the emulsification? Large drop of oil will be easily small droplets. I mean, so then it will be mycelium. So who will get it? Help to get mycelium? Bile. Bile is very important issue here. We will get mycelium. So mycelium is easily absorbed, and bile helps activate the lipase enzyme also to get more bile and to get more small droplets towards micelle, then fat will be easily digestible. You know, when we'll put fish oil especially for give energy and attractant also for fish. That's why we have to consider how it will be into small droplets. Longs and fatty acid is available in animal oil, so we have to consider. If we put fish oil, we have to consider bile to get it more micelle for easily digestible of the longs and fatty acid, to get it micelles to get more absorption of digested oil. You know, this is the beauty of fish, and even though it is a big challenge, if we get villi health good enough, so more nutrient will be easily digested and easily absorbed, and utilization of nutrient will be done in a good way. Main reason is that space to spaces, shorter bowels means weaker digestion. So we have to consider in what way nutrient will be absorbed and what type of help we need to get from feed additives and from functional feed additives, from the technological upgradation for feed additives to get more absorption and utilization of the nutrient. So this is very important area to get the villi health as expected, as good it is. You know, when the villi health will be good enough, then the nutrient will come through the villi to the bloodstream. So ultimately goal to get all the nutrient in the bloodstream, then it will go to the liver. Liver is the port, large port, very important organ of the body. So it's very important to get nutrient through the bloodstream to the liver, and then liver will get some works on nutrients to get the healthy performance of the fish. So from liver, you know, cholesterol is being produced, and that cholesterol contributes to production of bile. That is very important. So we have to consider fat also to get cholesterol, ultimately to produce bile. If we don't have enough bile, so how the fat will be utilized? So liver has some function. It will help to produce glucose, from glucose to glucosan, and liver has good activity to detoxification of poison and toxins, that may be endotoxin and exotoxin, and a very important issue is produces of bile. So this is an important area, and liver is good enough for fat soluble vitamins, vitamin A, D3E, and the essential amino acid also, especially methionine, and it has good function of hormones and enzymes. That is very important. So we have to consider in what way liver will be healthy, in what way villi length will be good enough and villi will be healthy. So nutrient absorption, what you have given in the feed formula, that should be digested, that should be absorbed, that should be utilized to get optimum and expected FCR. That is very important. And the enzyme, you know, in our fish feed formula, we have to incorporate some liquid enzymes. You know, why liquid enzyme, especially phytase enzyme and some other enzymes, protease enzymes. After extrusion, in the extrusion temperature is very high, so enzyme is protein, it may be denatured by the exposure of excessive heat. So we have to, after extrusion, we have to spray some enzymes. So if we spray enzymes, it should be enough substrate. If we spray protease enzymes, so we have to consider plant protein and other protein, because enzyme is there. If we don't put any enzyme in the formula, so we have to consider that easily digestible protein source will be added. So there is a correlation, what ingredient will be selected and how much enzymes and on what substance will be used for this enzyme. That is very critical issue we have to consider to get the good FCR through our feed. And it is also an issue, and we have to consider the temperature. Optimum enzymes, optimum temperature will be 35 to 40 degrees centigrade, and pH will be optimum 7. And within this range, we have to consider to get the good activity of the enzymes. Here is a sample feed formula for your understanding. In what way we are using local ingredient and important ingredients to get a formula. Here is a PABDA floating fish feed formula. You look into the formula, wheat powder and soybean meal, high pro, I mean a bit higher crude protein in a high pro soybean meal, and rapeseed cake, and fish meal. Local fish meal, there are some fishes which has been dried for using in the fish meal, but there is no standard CP. That's why it's in the always range, and it is sometimes adulterated with other things. So to get the least cost feed, some feed mill owners consider these things, but we are not actually recommending that. We would like to get the imported fish meal. And de-oil rice bran, this is available in our country. Bran oil is being used, I mean plant oil, bran oil, and de-oil rice bran also used as plant protein source. Limestone, dicalcium phosphate, and bran oil and palm oil, fish oil also, and Runion as feed additives for utilizing oil, because we have to consider 18 to 20 kgs of oil. So in what way this oil will be utilized, that's why we have to consider feed additives. And D-L-methionine, salt, and vitamin and mineral premix, and fish attractants also. In our country, there are some fish attractants. It is nothing but flavor. Same way for carb grower feed formula, same ingredient, especially a variation in the plant protein source and animal protein source towards least cost feed formulation. Same way for shrimp feed. Shrimp feed protein percentage is much higher, so that's why we have to consider poultry meal, dry fish meal, and a very important issue to eliminate toxins. That's a toxin binder. And in rainy season, we have to consider mole inhibitor. Liquid mole inhibitor is being used. Mole inhibitor is nothing but organic acids, and sometimes DDGS. Distilled dried grain with soluble, and sometimes corn gluten meal also used in our country, even though previously it was not used. Now it is being used within the limited amount to get it okay. If we use it, we have to consider the protease enzymes to get protein from the plant source, because plant source protein digestion is very poor. So this is the same way for pan gas. And for your consideration into the idea in what way ingredient is being selected, major items: maize in our country, wheat, rice polish, wheat imported and local production, maize imported and local production, rice polish it's available, soybean meal imported and local production, dual rice available, local, rapeseed local and imported a little bit, DDGS fully imported, corn gluten fully imported, dry fish local, fish meal fully imported, fish oil fully imported, soybean oil local and imported, bran oil it's locally available, and DCP and MCP, monocalcium phosphate, dicalcium phosphate imported. These are the imported and little bit local. So price cost all the times gonna up, because international market depends on the ingredients price. These days it is gone up, so it's very difficult part to get least cost fish formulation using a mixture of ingredients. And minor items, it's very common to all of you: vitamins and minerals, fat utilizer, then enzymes, especially protease enzymes, phytase enzymes, acidifier, I mean mold inhibitor, amino acid, pellet binder is very important issue. To get it bind, especially for sinking feed, it's very important how much it will be bind to get all the nutrient for a long time. Fish attractant, especially flavor, growth promoter, toxin binder to avoid mold and fungus, mold inhibitor, I mean propionic acids are being added to the formula. So good nutrition in animal production system is essential to economical production of healthy and high quality products. In fish farming, aquaculture, nutrition is a critical area. Typically represent approximately 50 to 60 of the variable production cost. So we have to consider the nutrition. Otherwise, we will not be able to produce more fish through a quality fish feed. So that's why we have to consider in what way our feed formula will be set up and the nutrient will be absorbed easily, and what are the feed additives we have to incorporate in the formula. That is the critical issue. We have to consider it again and again. Fish nutrition has advanced dramatically in recent years with the development of innovative functional feed additives. Nowadays, globally, there are some feed additives, they have some specific function. So we have to incorporate it in the formula to get it more utilization of the nutrient. Why should I get a nutrient drainage through feces? This is very important. And formulation of species-specific feeds support the growth of the aquaculture. This is very important. What species, what age, what environment, what season, and then formulation will be changed, ingredient will be changed. And on the basis of additive inclusion, formulation will be little bit changed. That is the critical area for formulating feed formula. Bangladesh has ranked 3rd position. I'm telling you again, so it has a good progress, and 5th in aqua production and 11th in marine. We are focusing for marine feed production because we have to consider technology and foreign investment. There is a huge scopes to be developed in marine fish production. Even though China has got good progress in marine fish production, so we have to follow the pioneer countries' technology and other a strategy in what way we can get it increased, especially for getting higher ranking globally. Bangladesh has created business environment. Good news for joint venture company. For fish feed production, fish processing, promoting fish hatchery, for producing quality fish fry, quality feed. Eventually, private sector has encouraged to the fish feed production and fish fry production and extension service also. So still there is a gap of technology and investment. So if we have more technology and investment, the production can reach double easily. So foreign investment are always welcome to develop Bangladesh fish production. And now we are actually wrap up the discussion. Fish feed formulas need proper selection of especially protein-based ingredients. Why I'm telling here protein-based ingredients? Because fish feed especially depends on high quality protein, which will be easily digestible. That's why a selection of protein base. If we select a lower quality protein, then we have to consider what type of feed additives we have to incorporate. That is a very important issue. Nutritionally rich quality ingredients are always needed for fish feed. And digestibility is the best parameter of plant and animal protein based ingredients are to be considered. This is very important, digestibility of plant and animal protein. And to reduce feed cost of plant and animal proteins, availability and price are to be considered. So if you want to get the least cost formulation, so what type of ingredients are available, we have to consider what are the limitations, how much anti-nutritional factors are there, what additives will be added, what are the challenges. We have to consider it, and we have to incorporate some feed additives to minimize it. To enhance digestibility and immunity, innovative functional feed additives, developed feed additives, new feed additives need to be used for further development of the feed formula and towards a good FCR, towards more fish production, even though other parameters depends on the total quality FCR, I mean pond culture and the technology, good quality, fish fry and other things depends on natural calamities, and these things are very important issue to have a good result towards good FCR. That's also be a considerable issue for getting good result in fish production. Thank you very much. I'm very happy to give you insight of the practices of our country for producing fish feed, selecting ingredients, and getting optimum result. Thank you.
Thank you so much, Mr. Akter, for your presentation. I believe our audience has learned a lot of information on aquaculture, Bangladesh aquaculture. Okay, from the feed formula for different kinds of fish and the mineral material using by the feed mill industry, and also promote the digestion and absorption of nutrients through adding the enzyme and some renew. So I think one question for you, Mr. Akter. The first is, does Bangladesh not use imported fish meal for shrimp feed? The fish meal from overseas for Bangladesh?
Yes, good questions. You know, very recent. We have to consider fish meal. Even though fish meal price is gone up, lower animal protein source was in Bangladesh meat and bone meal. You know, meat and bone meal. It was imported, but government has banned it due to some religious aspect. You know, our government always want to get it meat and bone from beef source. I mean cattle source, but they don't want to get it from pig source. I mean pork source. So that's why it has been banned. So we have to go to for fish meal, imported fish meal, or locally dry fish for getting as much lower as price we expected. And we have to consider other plant source. That's why we have to consider protease enzymes also. Nowadays, you know, protease enzyme is not good enough for sinking or pelleting feed if it is not thermostable. So we have to consider what protease enzyme is thermostable, then we can eat it. So there is a challenge whether it is thermostable. That's why after extrusion, after pelleting, we are using liquid enzyme.
Okay. I think the second question maybe after the webinar, you can communicate with Mr. Akter. We move to third question. What kind of enzyme needed for the fish? I think this is very well, right?
Yeah.
It is many kinds of enzyme.
Yeah, many. It's also good question. Many enzymes. You know, as a nutritionist, I have to point it out. What type of ingredient I am actually incorporating in the formula. So on the basis of that, how much it will be digestible. So to get more digested ingredient, we have to consider enzymes. So enzyme from phytase enzyme, protease enzyme, very important, and some non-starch polysaccharide enzyme, I mean Xylanase enzyme.
Wow, I think 4 more questions. Time is limited, so I think maybe later we will answer all the rest question in the afternoon. So thanks again for your presentation, Mr. Akter. Hopefully in the afternoon section, you can share more information during the Q&A section. Okay, so let's move into our fourth speaker, which is the last presentation for this morning section. He is Vice Chairman from Indonesia Aquaculture Society 2019 to 2024. coordinator for Aquaculture Nutrition Network Indonesia chapter. Also, he is a lecturer in Jakarta Technical University of Fisheries. His research interests are covering the development of intensive shrimp farming system, evaluation of nutritional requirements for shrimp and marine fish, and facilitate the evaluation of novel ingredient and feed supplement properties to enhance the growth and health status of aquatic animals. I guess some of the audience definitely know who is this. He is Dr. ROMI Noridia from Indonesia. So the topic will be the development of plant-based diet in aquatic feed. Okay, Dr. ROMI, now it's your time.
Thank you, Benjamin. Thank you. Can you hear my voice clearly?
Yes, very clear. Yeah.
All right.
Please
All right.
go
So
ahead.
I will share my screen right now. So hopefully everything is already there. All right. Good morning, ladies and gentlemen, participants for this very exciting webinar from LachanceGroup. So my presentation this morning is about the development of plant-based diet for aquafeed. So I'll start my presentation today with the, the graph that you already commonly. Hello?
Sorry to disturb you. Please enlarge your screen. You press the button. Yes.
I think it's already there. I mean, I already zoom it. Can you see it?
Okay, sorry. Please go ahead. Thanks.
So it's already zoomed, right?
Yes, please go ahead.
All right, thank you. So I'll start my presentation today with the, the common graph that we already seen in several, conference and webinar about the global aquaculture fisheries and also the aquaculture production system. If you look at the graph from the FAO, we know that the capture fisheries is tend to be stable, and the aquaculture is going to increase. So 2030, the World Health Organization and also the World Bank already estimate that more seafood supply will be coming from aquaculture. So this is our chance. This is our time for aquaculture to make the production become more productive and also more efficient. If we're talking about the feed for the total global aquaculture production system, we know that this is the the estimated global compound feed for the important species like carp, tilapia, catfish, salmon, trout, and also milkfish, and also vaname. We look at there, and the estimation, the need for the aqua feed in the future is around, right now is about 52.74 million tons in 2018. And this is just the estimation for the aqua feed need for, until 2025. From the 2000 and 2018 with the average production rate around 77.7% per year. So in 2025, in order to produce around 58.96 million tons, we're gonna need around 69.57 million tons. So aquafeed is a very good business sector in aquaculture. So how to expand the aquaculture production in the feed perspective? We need to decrease the reliance on the scarce and costly raw materials such as fish meal or fresh fish, and also more research in feed formulation and alternative ingredients to replace the dietary fish meal. So by doing this, we can hope that the production of aquaculture in the future will be more sustainable. What is the alternative for the fish meal? We know that from the previous speaker and also presentation, we need to, several alternative ingredients can be used to replace the fish meal. There are soybean meal, cottonseed meal, corn protein, canola meal, and other alternative protein. But important is for sustainability, the resource and also the environmental impact, social responsibility, and economic viability of these alternative ingredients must fulfill the sustainability criteria. If we look at the diet formulation in 1990, more animal meal is included in the feed formulation, like a 69%, and also 90%, only 90% plant protein source used in the diet formulation. Present time, with the massive research, we know that 53% of the plant protein source is already used in the diet formulation. It's only 31% of the animal meal used in the diet formulation. But in the future, we need to replace more. We need to substitute more the inclusion of the animal meal, and we expect that 69% of the plant-based protein source will be utilized to replace the animal meal. And in order to have a very good aquafeed diet in the future, we still need the novel ingredients and also the binders, to enhance the nutritional value of this aquatic feed that formulated with a plant-based protein source. We know that among the alternative protein source, soy protein is commonly used, but the wider use of the soy protein may be hindered by the negative effect associated with the presence of the anti-nutritional factors. We know that the proteinase inhibitors, there is a lectins, there is a phytic acid, saponin, phytoestrogen, antivitamins, and also phytosterol. There are several anti-nutritional factors that contain in the soy protein that can inhibit the utilization of the nutrient in the soy protein. So these anti-nutritional factors, they are responsible for the decreased growth performance, the decrease of the feed efficiency. Probably there is a histomorphological change in the distal intestine of some species of fish, and probably there is an alteration in the serum biochemistry. And consequently, further processing technique and proper blending to improve the nutritional quality of the soy protein or plant-based protein is needed. Right now, there are several advanced soy protein that are available in the market, and we know that two of them is there is an enzyme-treated soy, and then there is also fermented soy. The enzyme-treated soy, they use the combination of non-alcohol extraction process and also the enzymatic treatment to produce the advanced soy protein. And there is also the fermented soy. They use the microorganism to degrade the macromolecules to low molecular weight, so make them become more digestible and also easy to utilize and absorb by the aquatic organisms. Here, I just want to give you the comparison between the nutritional value of the soy protein, the conventional soy protein, and also the advanced soy protein that can be utilized in the aquafeed diet formulation. So if you can see the crude protein here, for the conventional soy protein, they are around 43%. We know the enzyme-treated soy with this enzymatic process, they can increase the nutritional value, especially in terms of the crude protein, up to 62%. And the fermentation process can also concentrate the protein up to 52.87%. We know that the moisture also decrease in the advanced soy protein compared to the conventional soy protein. And also the crude fat is being increased, and crude fiber is also somehow is a comparative. So if we just discuss first about the enzyme-treated soy, we know that the function and benefit of the enzyme-treated soy, they have a highest digestibility of protein and amino acid compared to other conventional product. And the protein was hydrolyzed to peptide before extraction, so it can be absorbed rapidly. They have extreme low anti-nutritional factors compared to the conventional soy protein. And the enzymatic process can remove the soy antigen and also the oligosaccharides, and most economical when replacing the conventional high protein source like a fish meal, blood plasma, or soy protein in the diet formulation. But if we're talking about the functional fermented soy protein, the function and benefit, we know that this fermentation process can also degrade the soy immunoreactivity, and they can have a lower levels of the anti-nutritional factors. They can decrease the molecular weight of the soy protein. They can improve the nutritional quality and also the fibrinolytic enzyme activity of the commercial soy protein, and also could partly prevent the alteration in the intestinal and also the liver morphologies in some species of fish. So in order to see what is the effectivity and how efficacy of this two advanced soy protein, we have a research to evaluate the fermented soy protein as the partial or total replacement of the soy protein, the conventional protein in the practical diet for Florida Pompano. So the objective of this research is to evaluate the several inclusion levels of the fermented soy protein to replace the conventional soy protein. We look at the growth performance, we look at the serum biochemical characteristic, and also we look at the intestinal and also the liver histological condition. So this is how we formulate the diet. So if you can see on the slide here, we partially and also completely replace the use of the soy protein. We would like to see whether this fermentation process can also have effect to the health condition of the silver pompano. We look at the diet formulation. So all the diet formulation have a comparable proximal analysis value. So here we have 4 treatments and 3 replicates. We put the 20 pompano per tank. and also 8-week growth trial and feeding strategy at 4 times daily. We adjust the ratio based on the percentage of body weight after 2-week sampling period, and we also measure the water quality to support the the carrying capacity of the recent growth trial. We can see that the water quality do not have any significant impact and all within the acceptable range for the pompano. And if you look at the growth trials, interestingly, there is statistically there is no effect to the growth performance compared to the when we partially and also completely replace the soy protein with the fermented soy protein. But we can see that in the histology, if we compare the conventional protein, there is a heavy inflammation in the intestinal condition of the pompano. If you look at the fish that feed with the fermented soy protein, the condition is more better compared to the conventional soy, and there is no heavy inflammation, and also there is no granulation compared to the basal diet, the control treatment. If you look at the liver condition, in the basal diet, in the control treatment, there is a heavy lipid accumulation in the liver, but when we feed them with the fermented soy protein, the condition of the liver is going better, and even I would like to say there is not so many alteration in the liver condition when we feed the fish with the advanced soy protein. In this case, in the fermented soy protein. So the summary for this research is the fermented soy produced by fermentation process using the Aspergillus oryzae and also the Bacillus may serve as a good source of the protein for the pompano. As the inclusion of the fermented soy to replace the soy protein increased, the diet were able to partially prevent the development of marked histological alteration in the liver and also distal intestine of the Florida pompano. So we need to, the further research, we need to evaluate in the long term culture period, probably from the stocking size until they reach the consumption size. So this is what we are doing right now. We still evaluate whether the fermented soy protein economically still provide a good economical cost for the farmers in order to increase also the productivity and also to have a very good profitability during the production system. So next, we also would like to evaluate the use of another advanced soy protein, what we call as the enzyme-treated soy. So the objective is to analyze the effect of the partial and also complete replacement of the dietary fish meal with various inclusion level of the enzyme-treated soy on the growth, also on the whole body composition, serum and enzyme activities, and also the histomorphological condition of the liver and also the distal intestine of the Florida pompano. So this is how we formulate the diet. If you can see in the slide, we have a 2 trial here. In the trial 1, we replace the use of the 15% of the fish meal, and we gradually decrease the inclusion level of the fish meal from 15%, 12%, 9%, also 6%. And the trial 2 was designed based on the results from the trial 1. So in the trial 2, we still have a decreasing of the fish meal inclusion levels from 12% to 6%, 3%, and the last diet, we completely replaced the use of the fish meal with the enzyme-treated soy here. So there's for the characteristic of this research, we have 2 series of the growth trial, 4 treatments and also 3 replicas per trial. We have also 20 pompano per tank, and we culture the pompano for 8 weeks with the feeding strategy 4 times daily, and also we measure the water quality as the supporting for the carrying capacity of the tanks during the growth trial. So again, we can see that for water quality, we are still within the acceptable range for the Florida pompano. But if we look at the final weight, this is interesting because a fish meal still have a good source for nutrient. We can see here as we decrease the inclusion level of the fish meal, we also observe there's a decreasing growth performance for the pompano, and also for the weight gain, and also for the thermal growth coefficient, and also for the FCR as we are trying to decrease the inclusion level of fish meal. We also do not see any efficiency in FCR, even when we try to replace the use of the fish meal with the advanced soy protein here. So from the trial 2, we also observe a consistency result. When we are trying to decrease the inclusion level of fish meal we also observe a decreasing in growth performance of the Florida pompano. So this is, the relationship between the final weight and also the thermal growth coefficient to the replacement level of the fish meal in the both trials. We can see here when we are trying to decrease the inclusion level of the fish meal, we also experience with the decreasing trend of the final growth and also the thermal growth coefficient in the trial. So this is the serum biochemistry, but also the nice thing, the most interesting is the use of the enzyme treated soy is almost similar with the fermented soy protein. Also do not cause any significant impact to the distal intestine and also the liver condition of the pompano. We can see that they are still better, but when we are trying to replace more in fish meal in the diet formulation, we also have the inflammation in the distal intestine of the pompano. So the summary for the second research, by using the advanced soy product like the enzyme treated soy, the continued reduction in fish meal levels will likely reduce the growth performance of the pompano. And in this study, the enzyme treated soy protein could be used to substitute dietary fish meal without compromising growth, nutritional, and also health status of the pompano. And further research, again, we still need to evaluate the efficacy and also the effectivity of this advanced soy protein in the commercial condition, meaning that we need to culture the fish from the stocking size until reach the consumption size in the long-term growth period to get the complete analysis for the use of the enzyme treated soy. So next, I also would like to discuss about another protein source, the DDGS. We know that this is the production of corn in the United States. They also produce the distillate grain solubles with this extraction process. After they get the ethanol and then the waste, the distillate grains, and then they distill it again, and then also get the what we call as the DDGS. So in order to evaluate the growth performance of the DDGS, we also have a research with the shrimp with the Lithopenes vename to partially replace the use of the soybean meal. We use the 3 levels of the DDGS, 5%, 10%, and also 15% inclusion levels of DDGS to replace the use of the soy protein in the diet formulation. And all diets were formulated to contain approximately 36% protein and also 6% lipid. And all experimental diets were produced at one of the commercial feed mill here in Indonesia. We produce it with the PT Suritani Pamuka, Java Group Aquafeed Research Center. And the growth diet were carried out in Batam, Indonesia. And this is in the right side, you can see the diet that produced by this feed mill by using several inclusion level of DDGS to partially replace the use of the soy protein in the diet formulation. And this is how we formulate the diet formulation. We use the 3 levels of the corn DDGS, 5%, 10%, and 15%. We also reduce the soy protein here, as you can see on the slide, from 25%, it's going down to 22.5, 20, and 17.5%. And we also supplement the lysine, because when we are trying to replace the soy protein with the corn DDGS, we know that the lysine cannot fulfill could not satisfy the requirement for the vaname. So we also make a supplementation with lysine here to just make the nutritional value comparable with the basal diet by utilize the 25% soy protein in the diet formulation. So this is the proximate composition of the diets containing various levels of distillate grain solubles, DDGS. You can see that we determine the diet with the 36%, and all the diet have a comparable value here. Also for the crude fat level, they have a comparable value among the dietary treatment. So the protein contents of whole shrimp body were analyzed by combusting according to the Dumas method. So this is the profile of the amino acid composition of the experimental diets. So if we look at the 3 limiting amino acid like a lysine, threonine, and also the methionine, we can see that this is they have a comparable value. And this is also interesting that when we're trying to replace and we substitute the use of the soy protein with the DDGS, the methionine level is going to increase. And also for the lysine, they have a comparable value here, more or less around 2% in the diet formulation. And also for the threonine, they also have a comparable value among the dietary treatment. The growth trial for this research, we have a 4 treatment and 10 replicates. We stocked the aquarium tank with the 15 shrimp with average initial weight of 1.04 gram. And we stock in the more or less around 100-liter aquarium tank for 52 days of the culture period. And the feeding strategy is a 4-time daily with assumed FCR around 1.5. And also the pH, dissolved oxygen. We measure all the physical quality, water quality, and also the water chemistry, but weekly and also daily for the physical water quality. And this is the result for the water quality, all still within the acceptable range for the Lithopenes vannamei. And this is for the response of the juvenile. We can see that almost all the treatment do not have any significant difference, meaning that when we're trying to reduce the use of the expensive ingredients like a soy protein with the cheap ingredients such as DDGS, they also do not have any significant impact to the growth. They still have a similar growth compared to the control treatment, as long as we blend them with the proper blending formulation, and also we supplement them with the proper nutrient. In this case, we know that using the DDGS, it will lack of the lysine, and when we supplement the lysine, then we have a comparable value here. So in the future, the crude protein level is not as a baseline, but how we determine the quality based on this specific minimum requirement, especially for vannamei. They are continuous feeder, and we need to fulfill the specific nutrient requirement in order to get the optimum productivity and also profitability in the shrimp culture production system. And this is the proximate composition of the whole body of the shrimp. Value represent the mean of the 10 replicates. We can see that in terms of the carcass, the body carcass, all the shrimp have a comparable value in terms of the crude protein, water content, fat, dry matter, and also the ash content in the diet formulation in the shrimp. As the conclusion for this research, under the condition of the present study, the DGS can be used up to 15% to partially replace the use of the soy protein and wheat flour without compromising the growth of the Pacific white shrimp Lithopenes vannamei. And economical in terms of the cost per unit of protein, DDGS is a very promising alternative to be used in the diet formulation. Further research need to focus on the mycotoxin issue and long-term effect on the use of the regular protein DDGS in shrimp culture system. We already done this in the long-term growth trial period, and we can see that there is a consistency result, that there is no effect to the growth of the vannamei when we're trying to culture the shrimp from the PL7 from 0.03 gram until they reach the consumption size around 25 gram in the pond culture condition. We already published this, and then it's easy to Google it to get the data for the long-term trial of this DDGS as the alternative protein to replace the use of the expensive protein. In this case, is a soy protein. So for the general conclusion for my presentation today, we know that Pompano has wide tolerance to the soy-based protein, and also under experimental condition, the use of the enzyme-treated soy, the fermented soy protein, and also the DGS for the shrimp could be used as an acceptable protein source to replace the fish meal and also another expensive ingredients such as the soy protein in the fish and also shrimp diet formulation. And the combination of the enzyme-treated soy with the 4% sweet hydrolyzed probably to replace the poultry meal also prevent the decrease in growth performance. So thank you for the opportunity to say my talk to LachanceGroup, and I'm ready to receive a question if you have. Thank you, and I give it back to Benjamin. Thank you.
Thanks so much for your presentation, Dr. Rumi. Okay, so this is a great talk. You have conducted a lot of research on the soybean meal, okay, like the replacement of the common soybean with the fermented soybean meal. Later, we have one question from our audience, and also replacement of fish meal with enzyme-treated soybean. Also, you have done some research on DDGS in shrimp feed, vannamei feed. Okay, thanks so much. Let us move to the question. Okay, the first question, I think Please explain the industry method for the production of fermented and enzyme soybean meal. Dr. Rumi, please.
Benjamin, say it again. What is the question?
Okay, for explain the industry method for produce fermented soybean meal and enzyme treated soybean meal.
You mean in the industrial level, right?
Yes,
so,
yes.
So right now, the use of the fermented soy already utilized by several commercial feed mill, because they know that this is what we know, that the several feed mill they produce the functional feed, and they use these ingredients as a part of the functional feed production system. So functional feed meaning that we need to include as functional ingredients or functional additive in order to enhance the growth performance and also the health condition of the aquatic organism. So this is make the functional feed difference compared to the conventional feed, Benjamin, because this the functional feed can have produce the superior growth performance, but we know that a farmer's always thinking about the cost, economical cost of the diet. But please look at the results. Please look at the results after we use the functional feed. We look at the survival rate, look at the productivity, and look at the the profitability. Then we can see that, oh, there is no significant increase in terms of the feed cost if we use the functional ingredients or functional additive in the diet formulation. Thank you, Benjamin.
Yes, yes, I fully agree. We must say the growth, the production. This is the most important part, because the farmers buy the feed, they want to get the high production. Okay, the second question, I think just now you mentioned you are going to do the further research on replacing soybean meal with DDGS. But at the meantime, you are facing the macrotoxin problem. So any solution to overcome the problem of macrotoxin?
Yeah, so the thing is with the proper production process right now, Benjamin, the mycotoxin is already going to decrease, and also for the better production system, we actually can minimize the mycotoxin problem with the proper storage system. We know that all the feed will facing a similar problem if we store them in the long period, Benjamin. Then if we're thinking about how to store the feed in the proper way, and also I think mycotoxin is not a big issue in the shrimp industry. But the thing is, yeah, there are a limitation also, Benjamin. There are a maximum inclusion level that can be used in the diet formulation. So in our research, 15% is okay, but probably more than 15%, you're gonna have a problem with the mycotoxin, or you have a problem with this. Also in your feed machine, probably you have a problem with a sticky diet or something like that. But yeah, please maintain the proper blending system, proper formulation in order to get optimal productivity by using a DDGS.
Yes, I see.
Yeah.
Because if I'm not wrong, I saw some papers, from online ICI papers. They mentioned the mycotoxin is very, very harmful for the hepatopancrease of shrimp. That's why this question, right. I ask this question from you. The hepatopancrease is very simple, but the most important gland for the shrimp. They have 4 types of cells, and easily and very fragile, easily to damaged by the mycotoxin. Okay, the third question is very long generally. Let me go to the question, try to understand the question first. Using some shrimp farm, when I will be benefit it together with it. I think later maybe this question we will reply to you after the webinar. And can change the meat color by using DDGS. So that means that when we use some DDGS in the feed, will the color of the shrimp or the fish, the meat color will be changed?
Yeah, so this is a very interesting question, Benjamin. We also have the organoleptic test here. When we try to use the more inclusion level of DDGS, yes, I'm talking about shrimp, Benjamin. The color, when we boil them, when we boil the shrimp, the reddish color is tend to be very fast compared to the conventional diet. So they are more reddish. They are more reddish. But if we trying to if you use a more inclusion of the DDGS, yes, the color is not quite good. But if you use the proper inclusion level, and then you can also increase the quality of the shrimp with the brightest color, with the reddish color on the shrimp when we try to boil the shrimp, and this is good. So the DDGS can actually decrease the economical cost, the formulation cost of the aquafeed, but at the same time you can also increase the quality of the shrimp by using the DDGS. But please not more than, don't use it in the more inclusion levels. So our recommendation is don't exceed the 15% inclusion level.
Okay, so I think you answered another question already, which is is it the DDGS can up to 15%? I think just now your answer already from your research. Okay, I think we have last question for you, Dr. ROMI.
All right.
Of course, a lot of questions will come in. Later we can communicate. The question is some farmers, okay, sometimes doing fermentation to their regular shrimp feed. Is this method helpful to increase the digestibility rate of feed? Okay, that means they buy the commercial feed, then they do some fermentation in their farm. Is it helpful for the digestibility?
I think the goal for the farmers doing this fermentation process in the farm with the commercial feed is to reduce the macromolecule, to degrade the macromolecule into micromolecules, so the nutrient it become more easy to be absorbed, utilized, and digest at the end of the when the shrimp feed the feed with this fermentation process. And also I think the fermentation process, other than to degrade the molecular weight, also to concentrate the protein level. So I think this is two main purpose of the farmers doing this fermentation process before they feed the shrimp in the farm. So they are really want to provide a good source of feed, and also when they provide ready to be used, ready to be digest, ready to be utilized, ready to absorb a diet, they can expect a good growth of the shrimp in the farm.
Okay, I fully agree, because in China we also have a lot of feed mill in that industry. They produce the commercial fermented feed for the shrimp, for the fish. Yeah.
Okay.
Some powder, some pellet. Okay. So later you can do more discussion with Dr. ROMI. Since we have 5 more minutes, so I have one more question, last question, Dr. ROMI. Okay.
Yeah.
I choose one question. So have you ever experienced a color discolor when using DDGS for substitute for shrimp feed? So have you ever experienced a color, sorry, I didn't get this point, the color discoloration when using DDGS for substituting for shrimp feed?
Oh, so yeah, I can see on the question and answer tabs. This is coming from Mr. Nilo Cordero, right? Have you experienced a color discoloration? So discoloration, I don't think I have experience on this discoloration, Benjamin, when I use the DDGS for substitute, because I normally make a recommendation to the feed mill, 5 to 10% DDGS can be used for to replace the use of the expensive protein like a soy protein. And I never had the experience on discoloration. So yeah, that's my answer.
Okay. Thank you so much, Dr. ROMI. Okay, hopefully we will see each other in future, face to face.
All right.
Okay.
Thank you, Dr. Benjamin. Thank you so much, everyone. Have a good one. Have a good day. Thank you.
Good day to you. Bye-bye. I think now we move to Dr. Mr. Akter. Mr. Akter. Mr. Akter. Mr. Akter, I think a few questions for you. Are you there? Okay, sorry, maybe we wait a minute. We can call him.
Hello.
Hey, Miss Akter. Okay.
Yes, yes, please, sir.
Okay.
Yes,
I
sir.
think I think a few questions for you. Let me check. 哪一个问题来着？ Give me a minute. I check because so
problem.
many questions.
No problem.
Above 30. Above 30. Let me check.
No problem.
Yeah.
I'm always ready for you.
Okay, thank you. And also thank you for our audience. Okay, the first is the first question, Mr. Akter, is
Yes,
dear Akter.
Yes,
Good
please.
to know that Bangladesh got the FAO higher ranking from fish production. Okay, if I'm not wrong, last year your production is around 4.6 million tons.
Right.
Okay, the fifth top 5 country.
Yeah.
Okay, is this solid in local market only, or it is export to international, to other countries?
Oh yeah, very good and inquisitive questions. You know, Bangladesh has got good progress in fish production. That's why FAO pointed it out in ranking. It is especially for inland water capture feed, and as well as culture feed for tilapia. And that's why in tilapia, it has ranked 4th in the world and 3rd in Asia. You know, there are so many ponds which are not used to culture tilapia and pangas scientifically. So it is increased vertically. You know, in the same area, they put more fish fry, and they took the technology and some sort of quality feed. And nowadays, a feed mill producing floating fish feed. You know, in tilapia, why there is a big change? In our country now, less amount of sinking feed in tilapia is used. Most of the feed for tilapia now, floating fish feed. So practicing feeding them, type of feed has already been changed from sinking to floating. So tilapia production has already been increased. And you know, some good quality brood fish has already been imported from other countries. So changes in good quality fish fry contributed a lot to this production. A few years ago, it was depression in inbreeding depression. So less production. Now in the same area, we are getting more increased production. So that's why FAO ranked for this culture of fish. It has been in the 3rd position in Asia. You know, it's a good news for tilapia.
Yeah. Yes. I think glad to hear that. Hopefully in future, maybe you can come to 4th or 3rd, please. Okay, let's move to the second question. The second question is it also the problem and the challenges for fish feed formula in Bangladesh? Okay, the utilization of local raw material. Utilization of local raw materials to use for local fish production, because the quantity and the quality of local produced raw material.
Yeah, very good question. I think it is from a nutritionist. You know, in Bangladesh, there is a bad practice because they are not following the government standard parameter. I have already given you the idea that government set up some parameters of crude protein percentage, fat percentage, and other percentage. So very important percentage is how much percentage of carbohydrate, I mean energy, how much percentage of fat, how much percent of CP. This is the standard set by our government, but we are not following this one to get it least cost feed formulation. So we are considering low quality raw materials which is available in Bangladesh, but we don't know nutritionally how it is okay for fish and how much digestible it is. And there are some adulteration also. In true sense, to be honest with you, there are some suppliers, they don't know what will be the quality for fish feed raw material. So adulteration is happening there, and storing capacity is not good enough. For an example, rice polish, rancidi, right? You know, pure rice polish, very rich in fat. Fat is very important. So fat will be easily digestible to get more energy. So we don't know how much it will be preserved and in what way it will be preserved, and within the stipulated time it should be used. We don't know, but supplier giving us as a low cost solution. So that's very important. If we consider the low cost ingredient, we have to consider what will be the feed additives to be incorporated in the feed formula to get the feed safe for fish. Thank you.
Yes, thanks for your sharing. I think there is plenty of the jobs for the researchers, for the experts like you need to be done in future. Okay, so let's move to the third question. The third question is, what are the basic considerations while formulating floating feed and sinking feed for carps?
Oh yes, good question. Because to get more fish production, especially for fish feed, nowadays in our country, what is being practiced, you know, when fish are very small, they're using some sort of powder, nursery powder, that is floating type powder. So when the fish is small, so floating of the fish feed is very important issue. So for floating fish feed, what should be considered? Considering issue is that low bulk density. So what type of ingredient will be used? That should be low bulk density. I mean, it will not be easily go to the bottom. So it will keep sometimes within the range of the water to feed it well by the fish. So this is very important issue, and it should be oil coated. So to use oil is very important issue here. For sinking feed, you know, for sinking feed, if the mixed culture of the fish, I mean, not so intensity of specific one species, then sinking feed is not good enough. So for sinking feed, some feed will eat from the top of the water, and some middle of the water, and some bottom of the water. So for sinking feed, if it is go down to the bottom, some fish will eat this fish. And third one considering issue is that for sinking feed, binding capacity and gelatinization, binding capacity and gelatinization of the carbohydrate is very important issue. And floating fish feed, oil coated, is very important issue. These two parameters are important issue to preparing floating and sinking feed, and the considering the ingredients also issue. The reason is that low bulk density ingredient will be used for floating fish feed.
Okay, thanks for your answer. One more question. The feed formulation based on the ideal protein is used to reduce the cost of diet for, and it's used to reduce the cost of diet and the environmental pollution in poultry feed. Okay. So why this method didn't apply in the fish feed formulation?
Oh, very, very good question, and environment friendly friend question. I appreciate him asking this type of question. You know, we are always for protecting our environment. Okay. In poultry, we are using some sort of enzymes, especially protease enzyme, to get all the proteins or best amount of protein will be digested. That is very issue. So for phytase enzyme, it is considering issue. If we don't utilize the phosphorus. So you know, phosphorus will be coming out through feces in the nature, and it's not good for environment. That is being practiced in poultry. So for fish feed, you know, if you don't use any protease enzyme, so protein, undigested protein will come out and it will be in the pond, and then, you know, amine, from ammonia, it will be converted into nitrite. Nitrite is very toxic, you know, and the bacteria will ferment it to get it nitrate. Nitrate is very safe. So why should I go for toxicity in the pond? So that should be the considering issue, to utilize a much amount of protein which will be in the feed, to avoid environmental pollution in the water, and to reduce the phosphorus, I mean phytic acid, to reduce the phytic acid. Phytase enzyme is, using phytase enzyme is the issue for protecting our environment. So that's why it is being practiced in the poultry feed. So I would suggest that, a good question, we should use phytase enzyme, protease enzyme, and sometimes considering the ingredient, we should use glycanase enzyme also. But there is a question, we should ask the supplier whether it is thermostable or not. If it is not thermostable, we can get it after pelletization or after extrusion. If it is thermostable, we can use it before pelletization or before extrusion. These are the critical area. Thank you.
Yes, I think Mr. Akter shared a lot of information about this question. Okay, I think one more question, Mr. Akter.
Yes, please.
As we know, Bangladesh start to farming the tiger prawn. Whether there is some vitamin farming, can you explain? Could you explain some shrimp farming in Bangladesh?
Yeah, yes.
Like the production, like the species, like the diseases and the feed, whatever, please.
Yes. A very good question. You know, especially for shrimp feed in the coastal area, they are producing it, and price is very high also. It's profitable business if there is no environmental risk or disease risk. You know, in our country from the very beginning, it was less prone to viral diseases. You know, less prone to viral disease. Day by day, it has increased to prone to viral diseases. So it's a big challenge to minimize the viral diseases. So that's why we are advising them, you have to consider the water condition, because, you know, high hot temperature is high, and the water depth of the water is not up to the mark, then automatically shrimp will be getting stress, and that can lead to their death also. That's why pond management is a big issue. Number one, we are promoting this pond management, and for avoiding viral disease. Nowadays, government has got some idea that it should be virus-free fry. So that's very important. Virus-free fingerling, that's very important. That is the priority issue. So number one challenge is pond condition. Farmers actually don't know how to manage pond, and how to manage a pond according to the variation of the temperature. That's very important issue here in Bangladesh. And number one, feed also. There are so many anti-nutritional factors. Some feed mill owners, they don't know, they don't have exact consultation with the appropriate experts to minimize the anti-nutritional factors. So all the times, shrimp are vulnerable to prone to diseases. So this is a problem, and the pond condition problem, and traceability, you know, traceability for developing export market. Government try to minimize the traceability, how it will be produced from production to suppliers and leading to export market. There is a chain, that change is monitored by our government authority as well as farmers got some instruction. These are the SOP. Otherwise, you wouldn't be able to export this shrimp and prawn. That's the considering issue. And nowadays, it is being increased to get quality shrimp and prawn production in our coastal area, and government supporting them financially and providing extension services, I mean technical services.
Okay, that's good. I think in future, between China and Bangladesh, we can communicate anytime.
Yes, communicate. I know. Yes.
Yeah, for the shrimp farming, you know, from extensive, intensive, super intensive, our density can up to 1,200 per square meter. So which is pretty high. The production per square meter is 20 kilo. It's incredible. Yes, super intensive. And I think thanks so much, Ms. Akter, for your drawing, sharing so much information. It's quite useful and helpful for ours, for all the audience. I think now we coming to the end of morning section. So again, big thanks to all the presenters and also audience. Our afternoon section will start from 2 p.m. Beijing time. Okay. Hopefully, we will see you again in this webinar. So enjoy your lunch and have a good rest.
Yeah.
See you. Thanks.
See you. Thank you all for enjoying the webinar and interacting with each other for the betterment of the industry for all of the countries who participated in the webinar. Thank you all.
Yes. Hopefully, we'll have will be helpful for the industry's sustainability. Okay, see you guys. Hi, ladies and gentlemen, glad to see you again. So for our first speakers in the afternoon, he is a doctor of philosophy of Zhejiang University in China. So he is a strategist research project coordinator in CPP, Indonesia, and he has published 5 papers. Let us welcome Dr. Rajiv Kumar to present his topic on benefits of plant protein. Dr. Rajiv, please.
Hi.
Hi, hello. Good evening, Dr. Rajiv.
Thanks. Can I share my screen?
Yes, go ahead.
Is it visible?
Yes.
Shall I start?
Yes, please.
Yeah. Hello, everyone. This is Rajiv. I'm representing CP Prima, and the topic for today is plant protein benefits in aquaculture. So a brief content, I'm going to talk introduction, then uses of plant protein in aquatic animals, then benefits and utilities of plant protein, and functional food amino acid. So in introduction part, I would like to emphasize on how the seafood and aquatic industry is increasing. Overall, if you see, the annual growth is about 8 to 9%. If we talk about 2018-19, but if I take the gradual increase and take example of one commodity, which is shrimp, you can see its continuous increase of 4.7% gradual increase. And if I just take example of Indonesia, you can see that shrimp is contributing almost 19%, and in terms of revenue or value, it's about 40%. So it's going to be more and more increase in the production, because it is generating revenue and the employment to the country and other countries too. So this growth of aquaculture has resulted in an increased need of a specialized compound of aquafeed. Aquafeed rely much on fish meal and fish oil for the supply of major essential nutrients such as essential amino acids, essential fatty acids, minerals, and attractants. The demand for fish meal and fish oil for aquaculture use is predicted to increase, consequently reducing its availability. So when the demand is increasing and it's a natural resource, so the supply is keep on reducing, or limited supply is there. Now come to the protein requirements for aquatic animal or animal in general. In order to meet the best growth performance of aquatic animals, it is very important for us to provide the indispensable amino acid IAA in sufficient quantities and in the right proportion. So far, scientists all over the world have found that there are 10 different kinds of IAA, which are phenylalanine, valine, isoleucine, methionine, leucine, tryptophan, lysine, threonine, arginine, and histidine. Conventional sources for protein substitutions in aquaculture. So there are, if we point out, there are three main protein sources which are used in feed, animal feed in general. It's animal nutrient sources, then plant nutrient sources, and microbial nutrient sources. So for decades, studies on fish meal replacements have been conducted, and scientists are going for the, for the insects, for animal, based replacements. And these studies assess not only the digestibility of feed and effects on growth with amino acid supplements, but also assess health status and flesh quality, that how the texture of the meat is, how is the increasing or decreasing the protein level in the meat, total energy, and also the cholesterol and creatinine level. If we look at the overall acceptance of plant-based protein and its sources, we can see from the graph, you can see worldwide and particularly of Indonesia, you can see there is gradual increase in the studies for last 20 years. And I have data for up to 2018, and you can see there is highest percentage of publications, which are on the oils, natural oils, or on plant proteins, or on herbs are going on. This data is only about the aquaculture. If we talk about country in particular, Indonesia, almost 46% of the farms, they use more or less herbal products. Come to India, there are many specific herbal extract based remedies for aquaculture and livestock which are coming in the market every day. China, especially after the ban on antibiotics in aquaculture, this has given ample chances for herbal extracts to mix in the feed or directly in the water to improve the immune system of aquatic animals and so the digestive system. This figure also demonstrates the popularity of herbs as an alternative and substitute of drugs in aquaculture and animal husbandry. Going to next, so plant-based ingredients. Plant-based sources are still the preferred alternatives for replacing fish meal in aquaculture feeds. Plant-based ingredients are used not only as a protein sources, but also for other nutritional and functional requirements, such as starch used as a binder. Soybean meal is a widely used plant protein source in aquaculture industry and is the most studied ingredient in terms of fish meal replacement. Focused research efforts are ongoing to access the suitability of other plant sources. So you can see from the table, all are soybean, and it has different protein percentage. These are the different sources, plant sources like soya concentrate, wheat gluten, corn gluten, soybean meal, rapeseed, sunflower, lupin, peas, fish meals, and it has different compositions of amino acids which are more or less competent to the existing fish meal. Now how we can correlate with the plant and the pathogen or the nutritional side, which I will talk a little bit. This is it's a theory of evolution, and on the right side I am trying to describe the different theories about the evolution. And when the pathogen like virus or bacteria, they mutate in terms of they mutate to survive, or it's a part of their evolution. In the same way, plants also go for evolution, and they have mutation in which they have the active ingredient or the ingredients which terpenes and sesquiterpenes. These are the ingredients which also develop and modify as per the evolution, and they are equally competent to go against or fight against these pathogens. So we work on this theory. Then come to why it's not going with one plant. You will see recently most of the herbal product or ingredients come in combination, that it is more than one type of sources are there. This is called theory of synergism. So it's not always 1 + 1 is 2, but 1 + 1 could be 11, 1 + 1 could be 111. So this also is the good concept, in which we see that many extracts which were found to have a specific activity by themselves, a blend of each extract may enhance the required outcome, like 1 + 1 is 11. Bioactivity synergism between different preparation or compound may improve the effect of a specific blend or extract. So we have to be careful. 1 + 1 could be 11. 1 + 1 could also be 0 or -1. So we have to find the right combination, and right combination which can give us the best result. So on the right side, we are describing the same, how the process occurs. Now this trying to show this graphics, that how it has the multiple benefits. When we follow the theory of synergism and the multiple effect which is happening inside the body, where body physiology also supports these natural ingredients to work in the most efficient way. So we are trying to show you from the different colored dots that it works as anti-stress response and anti-free radicals, anti-inflammatory, antiviral, antibacterial, and cellular signaling apoptosis up to that level. That's why there are reports that many herbs could have anti-cancer effect. Now come back to benefits in aquatic animals. So protein benefits. One of the main characteristics of aquatic animals is their use of amino acid as energy substrates. Why we eat, why animal consume food, all is to transform into energy. So the maximum or optimized energy, where you get the maximum level of energy, and then that energy converts into growth, and that gives the better FCR. So protein will reflect the anabolic phenomenon, growth of the fish or shrimp body. Protein requirements are the sum of the requirements for individual essential amino acids and the requirements for non-essential nitrogen. Some major benefits of protein in aquatic animal are improving growth performance, enhancing immunity, promoting the maturation and reproduction states. That's what we look for. I would like to show you some of our studies. where we see that how plant extracts the synergism, it helps against various pathogen. The model we are using is computerized in silico model, where we are showing you in this model that how it works, how the combination of natural ingredients, they work against AHP&D Vibrio bacteria, and it binds to the proteins which are supposed to be the like PA and PB, which are the active site, which of the toxin which causes mortality in shrimp, and how these active ingredients from the plant blend binds to them and make them inactive. In the second model, we would like to show you about the virus, which is it is foot and mouth disease virus, where we are showing the same how this protein is helping to bind the pathogenic sites, which are also protein based, to deactivate the virus. These are the different active components from the plants. We have some other publications which are also showing it affects on the white spot syndrome virus of shrimp and other pathogens. Now this one is about the protease. This requirement implies protein for maintaining the life-sustaining process like respiration, blood circulation, excretion, osmoregulation, and digestion movement. The maintenance requirement is the underlying metabolic needs of the organism under conditions allowing no growth, no weight change. All animals have relatively low yet absolute requirement for dietary protein to replace proteins lost in maintenance and metabolism. Here you can see that if we take example of katla or Labiorohita, which is rohu fish, the protein requirement in fingerlings or the babies are lower than the fry. Fries are more smaller, so smaller the fish, more requirements of protein are there, and it's more or less in all the species. These all are the freshwater fish. So maintenance requirement may be different, depends on the species and each growth. It is ranging from 0 to 5% of the body weight per day of diets containing either 25 to 35% of crude protein from casein to gelatin mixture. Protein requirement for growth can be expressed as a percentage of diet dry weight. The concentration of protein in the diet that produces maximum growth is the most used measure of protein required for growth of fish. The relative protein concentration required by fish or shrimp may be higher because fish need more protein for growth. Juvenile shrimp offer 32% protein diets, indicated requirement of about 46.4 gram DP per kg body weight. Shrimp offered 48% protein diet, which is generally in China. Indicated a protein requirement for maximum growth is 43.4 gram. I would like to show you one of the examples that when we started using from 2020, the plant ingredient in our feed, we can see that there is a continuous increase in the tonnage. We could get up to 20 ton per hectare. Survival rate is about 89% and lower FCR 1.24. Irrespective of the lower stocking density, you can see it's only 115, whereas in 2019 and 20 it was 130. We reduced it to 115 pieces per meter square, and also we can see that the harvesting size is lower, but because of the better survival and firm meat quality, we can get better productivity and also the tonnage. This is tilapia, where we can see that when we are mixing plant ingredient in the feed, we can get better tonnage and better survival, and so the FCR in both cage and in the tanks. This we can show you here on grass carp, where we can show that when we are adding plant ingredients in the feed, we can have better FCR 2.26 versus 1.27 or 0.97 at different concentration, and the survival rate also increases 92-91% compared to 80%. This I would like to show you a little bit on the animal side, on the quality of pork. And what we see, which is really interesting for me, is the cholesterol level is reduced while using the plant ingredients in the feed, and we get higher protein level and higher ash level, and total energy is and the fiber content increases, carbohydrate percentage increase. And also, for your reference, you can see the uric acid level also reduced 1.78 compared to control, which was 2.92. And we also had the blind test of the meat, boiling pork, and people, they like the meat which was grown on the plant ingredients. So on the reproduction, it is known that reproductive performance as well as egg quality in fish or shrimp is affected by several factors such as nutritional status, stress, egg over-ripening, genetics, age of broodstock, water quality, and environmental factors. Like there are multiple factors which can affect Then later in maturation period, muscle protein appears to provide the major energy source. The depletion of muscle protein was mostly matched in females by an increase in ovary protein, and in males by rapid secondary sexual development. So how it affects? I would like to show you one which trial was conducted in Vietnam by our colleague Dr. Khoa, and he found that the sperm count increased up to 25% after 3 weeks of consumption of essential oil based or plant based diet. These are the list of functional amino acids which is which are how they are significantly important for different species of fish. Here I would like to emphasize you on how we can have value added products using the plant, how we can utilize plant protein or plant ingredients. Recently I attended one conference in India, Aqua India, and this presentation is from the Vice President of MPDA, Marine Product Exports Development Agency, and this number 1 and number 2 gave much enlightened to me. First is a special feed. So this is called herb feed or herb shrimp. So from herb feed they produce herb shrimp, which is having a special ingredients which are combination of herbs, and it gives which reduces the uses of fish meal or other ingredients, and it is giving like lemongrass shrimp or olive oil or ginger shrimp, like that. So this is from the Japanese buyer who are trying to get exotic shrimp with this kind of addition. The continuation of that is the aroma or flavored shrimp. So it's the same way it is added, and herbal shrimp is produced. This generally is done by adding the special diet in shrimp for last 30 days of culture, where shrimp gets a kind of texture, aroma, and also the flavor. I think this is a new idea which is coming up, and this will boost the uses of plant protein in general in the shrimp and fish industry. This one more thing which I saw, that uses of plant-based products are helping to reduce the methane emissions. And one of the examples which was given by Dr. George Chamberlain was that red seaweed supplementation reduces enteric methane by over 80% in beef steers, or 2% in cow's diet can reduce methane emission by 80%. So these are the excellent uses and benefits of plant protein, which I wanted to share and discuss with you all. I would like to thank my company CP Prima for giving me this opportunity, and Longchang for this opportunity. Thanks a lot, Benjamin.
Thanks, Dr. Rajiv, for your presentation. So I think you introduced a lot of information on functional of proteins and functional amino acid. And also I heard you have one trial on the essential oil. So that's good. Later, maybe we can discuss a bit. So now we have a few questions, Dr. Rajiv. The first, I think, let me check. So the first, okay, give me a minute. So, let me check. So can we have a clarify? Okay, the first question. So can you please correlate the effect of plant protein percentage on shrimp acceptance? I think maybe the percentage is 30% or 40%, and also performance. This is from Ramnak in the Q&A box.
Thanks for the question. I could see that. So this is that our scientists and scientific team have been working for long. It's been over 10 years. So because we are based in Indonesia, we don't have Ph.D here. So we started working in Vietnam since 2011, and there we established the optimum dose, and which can reduce the possibilities of getting shrimp infected by Ph.D, and also it could give the best optimum growth and the color, because if this plant protein also enhances, or I would say it brings back the natural color of shrimp or texture. So you will feel while using this these products, shrimp have better texture and sweetness.
Okay. So another question is, as we all know, if too much soya protein in the feed, in the aquatic feed, especially for the shrimp, it may cause problems like interitis and the slow growth, and also maybe the low efficiency of the feed. So what's your opinion or comments on this and how to solve such kind of problem?
Yes, in most of these plant origin products have some anti-nutritional factors. So we have to develop which can balance it, which can neutralize the effect. And I believe most of the scientists or the feed formulators, they know it and they make it. So in general, I would not tell that because soybean has been used for a long time, soybean meal, and we don't see in our shrimp or fish there is enteritis due to soybean. I think this could happen maybe long back, but not now.
Oh, no. Okay, I see. So one more question. I saw in your presentation, you mentioned that EO is essential oil, right? For the pond guard or what, how effective it is for the shrimp farming? Because in China this year, Ph.D in south of China is very, very, very common. So frankly to say, we have lost south of China. We lost 80% of mud pond shrimp. Now we start restocking. Okay, this is very serious. So far, I think based on your presentation, I saw the Indonesia, the survival rate is pretty high. It's more than 80%, or some reach 86%. So
Right.
you share some critical and important things to us.
Well, I would say, that what we call it, a new normal standard operational procedure. So it's like a package I will share later with you, which our company has developed, where we go with the, the better pond preparation. We have to apply more lime. Then we have reservoir, where we have to sanitize the water, which before it was 30%, now we have increased to 33 to 35% water holding. And then on the culture side, we reduce the stocking density. If you know about Indonesia, before we used to stock 120 to 150 pieces per meter square. Now we reduced to 80 to 65. So it's low. And then we go for partial harvest. Partial harvest is to maintain the carrying capacity, so we don't overload the ponds. Then the best thing what we do here is siphoning. It's a kind of pond bottom cleaning. The same way how you clean the aquarium. When aquarium doesn't have the filter, we use the suction pipe and we clean the aquarium. In the same way, our farmers here do pond bottom cleaning. Before it was after a month or two. Now we started from second week, because we found that when the culture starts after 2 weeks, there will be some organic material settling down at the bottom, and that creates environment for EHP and for Vibrio. So that actually helps to take out. So we have to do more labor work, and above all, we have reduced the size of the pond. So now the average size you will find in Indonesia is almost like 2,000 to 3,000 square meter. Previously it used to be half hectare or 1 hectare. So this we have changed the management practice, and that is helping us. So it's complete. Of course, Pond Guard and EcoGuard are helping there. It's a complete package, I would say.
Yes, I fully agree, because we need the good pond preparation. We need to sterilize the water. We need the good quality of SPF, PL, and good management, and never overfeeding, good quality of the feed. So this is every point is also very, very important. Okay, so one more question.
Yes, please.
With plant or plant-based extract, is it possible to reduce various microbial disease in fishes? What are your views about control of ecto and endoparasite control?
Yeah. So again, when it comes to parasite, it's more difficult job, because it becomes more mechanical than immunity. Mostly we are focused on the viral and bacterial pathogens. We have less experience so far on the parasite, especially the external parasites. I guess this question is from India, where where we have to maintain more on the biosecurity side. We have to make sure that the water should not be contaminated, the water entering to the pond. And for things still, chemicals are working better than the plant, because a plant cannot be so strong and aggressive to kill the the parasite, which are generally bigger in size. So we have to plant is a kind of mild thing. So for the strong, this kind of killing the parasite, killing the big ectoparasites, we have to use chemicals or mechanical things. Like sometimes suppose for leaves, we from fresh water we put in the salty water, and then these lyses come out, and then you make it get rid of lice in tilapia or in carps. So still those methods are more productive, and we also have to think on the cost, because when we talk about fish, Mr. Benjamin, as you know, the cost of production of these fish when it carps are quite low. So if you suggest something expensive to the farmer, it may not fit to their budget. So
Yeah.
So we have to find the best way how to solve their issue. Maybe we will interact with Mr. Srinivas and Mr. Shivi, and we'll see how we can help them. So please be in contact with them, and we will work together to find the best solution for them if we could.
Okay, that's great. That's great. Yes, because for the farmers, for the farming, they need to make money. So I think we can give the best ROI to them. That is the best way for the farmers. And one more question also from Shivi. In one of trials, essential oil application to reduce fat and cholesterol evident. What do you suggest about usage of essential oil in salmon?
Again,
also from many.
I would say we don't have experience with salmon. It will be a new area. But what experience we are getting is on white shrimp, which is Vaname, then on tilapia, common carp, then on pangasius fish, then on pig and chicken. So here we see, there is significant reduction on uric acid level and creatinine level and on cholesterol, in the meat and also in the blood. So I feel, there are mechanisms which are working well with the plant ingredients which are able to reduce this. I guess it will be the same for salmon, but I don't have direct, data and experience with salmon.
Okay, I think many you can talk to Dr. Javier Gonzalez. I think he has a lot of experience about the salmon, about the trout. He's from Chile. So later you can communicate through email or through WhatsApp. Okay, so I think that's all for Dr. Rajiv. Thanks for your sharing, and thanks for your patiently answer our questions. See you next time.
Sure.
Okay.
Thank you.
Welcome to China.
Soon.
Yeah. We come and visit us. Thanks. So now we move to our sixth speakers. He has been working in aquaculture industry for 15 years. Used to work in Tongwei Group, which is top 3 company in China, for 8 years as feed formulator. He is technical director of Shandong LachanceGroup, and he has published 6 papers. And please welcome Dr. Zhang Wei to share the topic, application of bile acid in the low fish meal diet of shrimp. Dr. Zhang, now is your time.
Okay. Can you open my video?
So, yeah, okay. Give me a minute. Okay, now?
Okay.
Okay. Okay.
I will share my screen.
Yes.
Okay.
Yes, please go ahead.
Okay. Hello, everyone. Today, the topic of my speech is the application of bile acid in the low fish meal diet of shrimp. The speech divided into 3 parts. The first part is reason and problem of fish meal substitution. This is why bile acid are added to reduce some of the negative effects of post replacement. The second part is the mechanism of bile acid. In this part, we will know what is bile acid and how does it work in animal body. The third part is study on bile acid in low fish meal diet of shrimp. In this part, we will learn more about the benefit of adding biocide to shrimp in low fish meal diet. Let's get to the topic. The first part is reason and the problems of fish meal substitution. Fish meal is an important animal protein raw material in shrimp feed. It has many advantages, such as providing high quality protein, balanced amino acid, low anti-nutritional factors, high micronutrients, high health, high digestibility. Estimated global use of fish meal within compound aquafeed. A quarter of fish meal is consumed in shrimp feed. With the growth of shrimp feed, the demand for fish meal will further increase. In recent years, global fish meal production has declined from 5.6 million tons at the peak to less than 4 million tons, has dropped 33%. However, the whole trend of fish price is rising due to the decline of global fishery resource and growth in aquaculture increase demand for fish meal. Rising fish meal price lead to high feed cost. How to effectively deal with this situation? Therefore, we must reduce the amount of fish meal used in feed, thereby reducing the dependence on fish meal and the cost of formulation. Traditional fish meal replacement method is using the other protein source and supplementation with limited amino acid. Diets must be developed based on substitutability, and plant protein source could replace the amino meals, especially fish meal. This plant protein include soybean meal, cottonseed meal, corn protein, canola meal, and so on. In the future, the proportion of plant protein in feed formulation will increase from 53% to 69%, while fish meal will decrease from 31% to 10%. What problems with the fish meal substitution? Take soybean as an example. Soybean is an important source of plant protein for human and animals. It contains 20 to 25% oil and 40 to 48% protein. It's rich in vitamins, trace elements, and balanced in amino acid. However, soybean contains various anti-nutritional factors, such as protease inhibitors, lectins, and phytic acid, which can reduce the palatability and hinder the absorption of nutrition. Plant lectins can destroy the IgA produced in the intestinal, which damage the immune system and reduce immunity of the animal body. There is a lot of researchers on fish meal substitution, but the substitution is not a simple matter, and there are many factors and need to consider. Therefore, a comprehensive replacement strategy for fish meal is urgent needed. What are the negative problems of fish meal substitution? First, metabolism of various nutrients are suppressed in shrimp feed. Feed is a low fish meal diet. Glucose, triglyceride, and the total bile metabolism showed a significant downward trend. Second, immune response was suppressed in shrimp feed, the low fish meal diet, with the increase of fish meal replacement amount. The region of hepatopancrease and intestinal tract aggravated, and the health index decreased significantly. Third, induced enteritis, such as increased expression TNF-alpha, destruct tight junction protein, increase intercellular space, and makes the gut lose its barrier function, shorten of intestinal villi and thin of the intestinal wall. Exactly mechanism is not clear, but TNF-R is associated to MRCK. So do we have a feed active which could improve the lipid metabolism, immune response, and reduce intolerance of shrimp? The answer is to add bile acid to the feed. Bile acid plays an important role in digestion and absorption. It can serve as a critical single molecule in regulate immune response. It's also a major regulator of the gut microbiota. Dietary fish meal alternative often disturb the bile acid status in fish, result in either increased or decreased intestinal response and bile acid synthesis. It may be migrated by processing of fish meal alternative as well as the dietary inclusion of some bile acid. Although bile acid is traditionally regarded as unsynthetic in crustacean, a few recent studies has revealed the critical role of bile acid in the diet of shrimp. What's the bile acid? It's produced in liver with the action of 7-alpha hydroxylase, which with cholesterol is used as a raw material, which can regulate the metabolism of glucose and lipids, protect the liver, and regulate the balance of intestinal flora. Bile acid, which widely used in the treatment of human liver metabolic disease. How does bile acid work? Promoting fat digestion and absorption is the basic function of bile acid. This process is divided into 3 stages: emulsification, digestion, and absorption. In the emulsification stage, bile acid contains lipophilic group, which can emulsify large fat particles into small fat droplets. In the digestion stage, bile acid can increase the activity of lipids, change the spatial structure of lipids, expose the catalytic group, and decompose fat into monoglyceride and free fatty acid. In the absorption stage, acid can and free fatty acid form mixed many cells that are recognized by the villi of the small intestine and into the intestine lumen. Then free fatty acid, protein, and triglycerides form chylomicrons and into the blood circulation to complete the entire process of fat absorption. In recent years, studies have shown that acid also play a role in maintaining intestinal barrier function. Acid as TGR5 receptor can activate intestinal stem cell and promote the differentiation into intestinal villi cell. Stem cell maintains homeostatic renewal, injury-induced regeneration. We can summarize the function of acid: lipid digestion, absorption of fat-soluble nutrients, regulation of cholesterol level, and gut microbiota. Through the study of acid in shrimp, low fish meal diet, we can understand the role of acid in diet. Three also nutrients and isolipidic diet were formulated in this study. A positive control diet with 25% fish meal, a negative control diet with 15 fish meal, and 15% fish meal diet supplemented with acid. Fish meal was replaced by soy protein concentrate in this study. Low fish meal diet were supplemented with micro nutrients such as amino acid, vitamin, and materials to obtain a similar nutrition profile to high fish meal. Acid purity 98% was provided by Shandong Longchang Animal Health Products Company Limited, Jinan, China. Formulation and approximate analysis of this diet were present in left table. Shrimp and experiment condition, the average internal weight of shrimp is 0.3 gram. For receptors per treatment, feed 3 times a day. The breeding cycle is 8 weeks. The water quality is the most suitable for shrimp. Let me see the result. The final body weight, WG and FE, were higher in shrimp fed the diet high fish meal than those fed the diet low fish meal, and there was no difference between shrimp fed the diet low fish meal with acid. Survival of shrimp was similar among the 3 groups. Previous research relieved the plant protein often disturbed the acid status and reduced fish productivity. Anti-nutritional factors and high molecular protein are believed to contribute to it. This situation could be mitigated by the dietary inclusion of some bile acid. It was shown that supplementation of bile acid to the raw fish meal that promotes the growth of shrimp. Raw fish meal suppressed the hepatopancrease mRNA level of SOD and inhibitor of apoptosis protein. Dietary supplementation of bile acid in a raw fish meal diet enhanced the expression of SOD, CAT, and relish in hepatopancrease. Even show higher CAT expression compared to shrimp feed that high fish meal. There are no difference in the expression of Xbox binding protein 1 and capsid among the CIS3 group. Intestinal immune response. Shrimp feed the diet raw fish meal showed higher XBP1 expression compared to those feed diet high fish meal. Dietary supplementation of bile acid in a raw fish meal diet enhanced the expression of SOD, CAT, and relish in the midgut. The mRNA level of SOD and the CAT were higher and turbo was lower in shrimp feed the diet raw fish meal with bile acid compared to the shrimp feed the diet high fish meal. There are no difference in the expression of ERK among the three groups. Dietary bile acid supplementation upregulated the SOD and the CAT expression in the hepatopancrease and the intestinal, which indicated that bile acid supplementation enhanced the antioxidative ability of
Sorry, I think the internet got some problem. Wait a minute. I think just now we have one question. Maybe I take this time to explain to Doctor. Let me check the questions for the wet faces. So from Dr. Marsden, he has one question: what's the solution for wet faces? In my opinion, there is 4 reasons for the wet faces. If I'm not wrong, this is for the shrimp, right? Okay, 4 reasons. The first, algae crash. You know, once algae crash, then they will produce a toxin release toxin into the water. Okay, once your shrimp take such kind of toxin, will damage entire absolute cells from hepatopancrease and also the intestine, will cause wet faces syndrome. Okay, the second is based on our chief expert in China, Crustacean Association of China. He recently he has conducted one project on the raw material. Based on his research, there is 2 kinds of fatty acid. Okay, these 2 kinds of fatty acid, once your shrimp take such kinds of fatty acid, will cause the the disorder of the microbiota in the intestine. Then finally will cause the wet faces. hopefully we will see this paper soon. Okay, you can pay attention and search. Of course, you know the weather change, the stress also will affect the shrimp. Okay, so later I explain the rest too. Dr. Zhang.
Okay, sorry,
Okay.
just
Okay.
a little thought.
Okay, it's okay. So take your time. So the third is for the Vibrio, and EHP also will cause wet faces syndrome. Okay, Dr. Zhang, I pass to you. So
Okay.
please go ahead. Yes.
I will. Okay, everything is okay. Intestinal immune response. Shrimp feed the diet low. Fish meal show the higher XPP1 expression compared to the zoos feed diet high fish meal. Dietary supplementation of bile acid in a low fish meal diet enhanced the expression of SOD, CAT, and relish in the midgut. The mRNA levels of SOD and CAT were higher, and tuber was lower in shrimp feed the diet low fish meal with bile acid compared to shrimp feed the diet high fish meal. There was no difference in the expression of ERK among the three groups. Dietary bile acid supplementation upregulates the SOD and CAT expression in the hepatopancrease and intestinal, which include that the bile acid supplementation enhanced the anti-oxidative ability of shrimp. Dietary supplementation of bile acid in low fish meal diet reduced the ER stress in this study. Okay, hepatopancrease and intestinal histology. B cell was similar and R cell was fewer in shrimp feed low fish meal with bile acid compared with shrimp feed the other diets. Intestinal flows hair was shorter in shrimp feed the diet low fish meal compared to those feed the diet high fish meal and low fish meal with bile acid. TME analysis demonstrated that the midgut suffer more serious danger in shrimp feed the diet low fish meal, especially for ER, which was obviously swollen. The ER was back to the health after dietary supplementation with bile acid. Hepatopancrease B cell is the secretory cell, and the R cell is for storing nutrients. Fewer R cell in the hepatopancrease of shrimp feed low fish meal with bile acid indicated that nutrient storage was decreased. Nutrients may be fully mobilized to support the max growth of shrimp. The low intestinal fold, hair and swollen ER indicated the shrimp feed the diet low fish meal showed obvious intestinal damage in this study. The damage of the midgut would affect the digestion and absorption of nutrients and may be the main cause of the compressed growth performance. This result was similar as previous study. All proved that the high dietary plant protein inclusion would impair the intestinal health. of shrimp and fish. Dietary supplementation of bile seed can reduce the mid-gut damage in this study. Now, let me see the intestinal microbiota. As shown, the Shannon index was here in shrimp fed diet, high fish meal and low fish meal with bile seed, than those fed the diet low fish meal. And the Simpson index was lower in shrimp fed diet, high fish meal and low fish meal with bile seed than those fed the diet low fish meal, which indicates a lower bacterial diversity in group low fish meal. Dietary supplementation of bile seed in a low fish meal diet increased the community richness, such as CHO1 and ACE of shrimp. There are no difference in odors, numbers, and coverage among the three groups. Anyway, undesirable intestinal microbiota communities are induced by high plant protein. Let me see this part. The analysis of PCOA showed that the first two principal compounds explained 45.9% of the variation, and there was clear separation between the three groups. The relative abundance of gut microbiota in each group at the phylum level is shown in Figure C. The dominant phylum in three groups was proteobacteria, bacterial additives, cyanobacteria, and actinobacteria. The relative abundance of proteinobacteria was exceeded 80% in all groups. The relative abundance of gut microbiota at the genus level is shown in Figure D. The dominant genus in three groups were Vibrio, Regeria, Sima, and the Halosyncytibacter. A notable microbiota feature of shrimp fed diet low fish meal is the higher Vibrio abundance, more than 60%. Vibrio is a kind of typical harmful genera. The present result indicates a high Vibrio abundance may associated with the intestinal inflammation induced by plant protein. Dietary supplementation of bile seed may reduce intestinal damage by reduce the abundance of Vibrio. Regeria is involved in protein utilization. The higher Regeria abundance may associate with the growth promoting effect of bile seed. It's also regarded as a kind of probiotic in shrimp. The higher abundance of Regeria in shrimp-fed diet, low fish meal with bile seed may be beneficial to the intestinal health of shrimp. As shown in figure, dietary supplementation of bile seed in low fish meal diet significantly decreased the relative abundance of proteobacteria. Dietary with bile seed supplementation also increased the relative abundance of data bacteria. Besides the relative abundance cyanobacteria, Dada bacteria was higher in shrimp feed diet low fish meal with bile seed compared to shrimp feed diet high fish meal. Significantly, feature of top 15 genera were understood in this study, as shown in figure. 17 genera were significantly affected by diet treatment. 14 of them belonged to Proteobacteria. 3 of them belong to cyanobacteria.
Okay, I think his internet got some problem again. So let me explain more about wet faces diseases. From last October until now in the East Asia, it's very serious. Of course, in India also very common. So the 4 reasons, the first is Vibrio, the second is the EHP. Okay, Dr. Zhang.
Okay, sorry, I'm out online.
Yeah,
But
please.
I'm in.
Okay, please go ahead.
Sorry.
It's okay, it's okay.
Okay. Let me see this part. 14 of them belong to Proteobacteria. 3 of them belong to cyanobacteria. This relative abundance of Vibrio was here in shrimp feed diet low fish meal. Then those feed high fish meal and low fish meal with bile seed. The relative abundance of Vibrio was lower in shrimp feed diet low fish meal than those feed low fish meal with bile seed. These results indicate that low dietary fish meal level increased the harmful bacteria genera risk and decreased the microbiota diversity. Dietary supplementation of bile seed could decrease the harmful bacteria abundance and improve the microbiota diversity. Okay, let me say that this part, the bile seed are receptors of TGR5 and FXR, which can inhibit the NF-κB single pathway, thereby reduce the expression of pro-inflammatory factors in intestinal cell, thereby inhibiting enteritis and maintaining the function of intestinal barrier. Conclusion, low dietary fish meal, 15% decreased the weight gain, impaired the immune ability, induced the intestinal damage, and disturbed the intestinal microbiota of vitamin. Dietary supplementation of 0.1% bile seed in low fish meal diet can enhance the antioxidative ability, reduce the ER stress, improve the microbiota diversity, and decrease the harmful bacterial abundance of vitamin. Many studies shows that bile acid is also good for fish and benefit the growth, lipid metabolism, and composition of gut microbiota, but dependent on fish species, bile acid level. Our partner are located in more than 40 countries around the world. Our company is committed to creating wealth for 6,000 feed companies and 10 million farmers. Looking forward to cooperating with you sincerely. Thank you. That's all.
Dr. Zhang, thanks for your presentation. Yeah,
Okay.
I think our audience has learned a lot of information on bile acid and the application, because the bile acid is quite new to the aquaculture industry, I'm sure, as an innovative feed additive. Of course, glad to see LachanceGroup has done plenty of basic research on bile acid. Yeah. You shared the improve the immunity, the correlation between bile acid and microbiota. I think you answered one of the question already. Of course, just now you mentioned bile acid also can prevent or control the inflammation of intestine. Okay, that's good. I think later Prof. Orapint Jintasataporn also will share some knowledge on inheritors of shrimp. Okay, so a few questions for you. Okay, the first is how does bile acid help regulate the gut microbiota? I think you answer already, because through your presentation I saw that slide. Okay, so the second is how much bile acid should be added in the shrimp feed? What's the dosage? Let's say per times, how many grams or how many kilos, please?
We recommend the addition amount of 800 to 1,000 grams per ton, which will be adjusted according to different breeding stage.
Okay, so that means 800 grams to 1 kilogram per ton.
Yes.
Okay, yeah. I see. Move to the second question. What's the benefit of bile acid? Maybe you give us some summary, because you present a lot of information on bile acid. Yeah.
Yeah. Bile acid can improve fat utilization and reduce 0.02 to 0.05 FCR, reduce the stress price and the occurrence of disease, thereby increase the survival rate by 5 to 9%, and and increase weight gain, great benefit for feed mill and farmers.
Okay. That's great. 5 to 8%, 5 to 9% is quite a lot. Yeah. And I think no more question. Thanks for your presentation again. If our audience have any questions you want to know about the details about the bile acid, you can contact Dr. Zhang directly, since he is a technical director in LachanceGroup. Okay, thanks again. Let's move to our seventh speakers. So he is holding postdoc from Israel, Norway, and Spain. He is professor of fish nutrition and aquaculture, University of Kukurova of Turkey, and also he is the founder of Aquafeed. Okay, his research field is very broad, from fish nutrition to feed quality, lipid, fatty acid and metabolism. I think today many experts mentioned this word. Okay, macronutrients in aquafeed, feed quality and stress resistance. Over past 25 years, he has been working on marine fish and shrimp larva nutrition. He has published more than 100 papers. So this is the second speaker, published so many papers. I would like to invite Professor Tufan Eroldoğan to present his topic on challenge accepted fish meal-free diet in aquaculture, which is similar to Dr. Zhang, I think. Okay, so Professor Tufan, please, now it's your time.
Oh, thank you very much, Dr. Bai. I'm really happy to share some knowledge that we have in Europe, particularly in Turkey, with respect to replacement of fish meal in the diet for particular species. So actually the title is quite sharp, but I know that it's quite sharp, but I think we had some challenges and we overcome some issues in this respect. So that's why I choose this title to charm some people in the audience in the webinar. So, but we will discuss it later on. So my presentation will have at least 5 small topics, and then we are going to talk in details in these 5 parts. So let me start with the global macro trends, which is very common all around the world, actually, not only in Europe, but in Asia. So globally, we have some global macro trends, which can be population growth and health, exploitation of resources, and growing middle class and aging problem, and as well as the climate change, which is really hot topic at the moment all around the world. By touching upon each part, let's say global population is growing, which is expecting to reach 10 billion people by 2050. So I will talk about in details in my next slide on this regard. And also the global fisheries are to a large extent fully explored. And this is very problematic for the, let's say, fish meal resources, which we use in aqua feed and also poultry and some other, animal industry. And the another issue, which is called a macro trend recently, is a middle class, which is growing in large emerging markets and allowing more people to eat different and more nutritious and protein-rich foods like fish and, let's say, meat and eggs. So that's another issue which we are going to and challenging in due time. And consumption of high quality proteins for the old people is very important for their health. So it is quite a combined trends with respect to all these 6 trends together with for the animal nutrition and other issues. And also, of course, the climate change is greatest environmental concern and challenges to the world has ever faced recently. So concerns about climate change are, of course, influencing the dietary choices. But let's start with the population growth, which is a very common graph you may see from all the conferences. But we do know that, or all the let's say, aqua world in data, UN, FAO, OECD, present these kind of graphs. And we know that we are going to reach around 9 or 10 billion by 2050. So, of course, it's going to be food insecure people, which will be around 1.3 billion in due time. So that will be really case. Actually, if you ask me personally, we do have enough food in the world, but in equality, distribution of food is not equal and not fair. So that's why I think kind of an activist talking, it is quite different angle, but let's jump into my presentation because we are planning to produce more and more because of this population growth. But this is another challenging part for humankind, actually. So in the last 50 years, global meat consumption has increased around 70%, increased from 70 million to 310 million. So which means that we are going to need 70% more protein for available today. So that's really important things since we produce fish and animals. So we produce protein. So we need these 70% more proteins. So the new edition of State of World Fisheries and Aquaculture has launched in Lisbon just 2.5 weeks ago. And in this context, new FAO concept of blue transformation will align with European Union Sustainable Development Goals. So as aquaculture producer and feed producers, we have to take into consideration of these European Union Sustainable Development Goals. So this sustainable aquaculture expansion and intensification to support global food security targets and satisfy the global demand for nutritious aqua food is going to be very, very important things. So, but we need to see how we can going to increase the production with respect to this, let's say, blue transformation. So with this respect, global aquaculture production, as you can see in here, from 1907 to 2020, there is a big, let's say, increase during this time. And the main species group that contribute to the top 75% of aquaculture production in 2020 include seaweeds, carp, bivalves, tilapias, and shrimps. So the freshwater fish and species account almost 80% of global edible aquaculture volume. So which then we have to see the projection of 2030. So we do know that increased role of aquaculture for sure. So on the basis of assumption used and launched by FAO in Lisbon just 3 weeks ago, total fisheries and aquaculture production is expected to expand further and reach around 200 million tons in 2030. And the contribution of aquaculture right now is about 41%, and it's expected to reach around 56%. So we need definitely more feed and ingredients for the future dimension of the growth. So this is another graph that explains which kind of species that we mainly produce. As I explained to you before, particularly aquatic algae, and afterwards it comes with the fin fish and cultured in freshwater and crustaceans and so on. I will not touch upon this detail. You may find in FAO webpage of these graphs. And this is also another one which is very important because of the fact that even though we have we do not have enough fish meal at the moment, we also produce some fish meal from our byproducts, which I'm going to touch in my later slides. So I will just skip this one as well. But this is a big scenario that if in 2022 we reach around 122 million tons and we are expecting to reach 200 million tons in 2030, so then we have to, in a way, consider how we are going to do it, because in our hand we have 5 million tons fish meal oil from the fisheries and 1 million tons fish oil from the again the harvest from the oceans, and of course 2 or 3 million from the byproducts and so on. But what we do, the growth of contribution aquaculture will follow like this. We catch fish from the nature, produce a diet, and then produce fish. But it's not going to be sustainable if we are going to in this way. So that's why we need to have a look which kind of things as a nutritionist or as a scientist or as an industry produce some additives or produce some alternative raw materials for aquaculture, all right? So far, concerned research efforts have contributed towards the reduction of fish meal and oil. For sure, we did really good job. But in aquaculture, whether you produce shrimp or fish, there is no single feed, all species. As you can see on the left side, the carp, shrimp, tilapia has a different combination, quantity requirements change within a species. So also the raw materials, that are used in the formula has changed according to species. Like here, you can see that the difference between 2015 to 2025. This is an excellent article written by Xue et al. In 2090. And as you can see here that the feed carb is just only contains the formula of, in their feed, it's only 1% fish meal. And in marine shrimps, it's around 12 or 10% right now. So on the right side or left side, I don't know, I mixed it, but on my right side, raw materials in formula vary with species. So if we consider sustainable growth in aquaculture and feed, we cannot consider as one solution to all species. So it's quite changed, and it must be how to adapt on species specific basis. So this is one concern that I will again push myself to present something. And of course, aquaculture sustainability is going to have some challenges. It can be a biological, it can be an environmental, it can be an occupation of cost areas, and it can be a consumer's perspective in terms of food safety, food quality, and ethical farming standards. But for sure that optimal nutrition is the key to several of these challenges: biological, environmental, consumer, and occupation of coastal areas. All right. So therefore, agriculture, including aquaculture in 21st century, faces multiple challenges, which we know. It has to produce more food and fiber to feed a growing population with a smaller rural labor forces. And secondly, the industry contributes to overall development in many agriculture deepened developing countries. So then we have to consider these issues particularly and definitely in these kind of countries. And we have to adopt more efficient and sustainable production methods and adapt to climate change, for sure. But then we have another challenge. Are we going to feed the world, or are we going to feed the fish? So that is very, very important, aligned with the climate change and so on. So far, according to RoboBank infographic, as seen here, regarding with how to feed the world 2050, actually it is published in 2016 or something, but we achieved something in terms of, let's say, overfishing and other sustainability issues in the diets, and also other things which we really contribute. For instance, we really contribute aquaculture in terms of protein production globally. However, we know that we have not done enough, and that we must do more, better, and faster for the future. Okay, so let's have a look, feed production by species. So we are only contributing for the food industry in the world around 4.2%, which is account 51.4 million tons. So the poultry and pig and cattle production is really, really high. And globally, we produce 1,235 million metric tons global the global feed production, and the global feed production increased every year around 2.3% according to Alltech Global Feed Survey. So let's keep in mind this one. So how we are going to contribute our ingredient evaluation, how we are going to take into account raw material quality. So basically, we have 6 or more, but this is a general, raw material quality issues. The one is sustainability for the future growth and for the sustainability of the production. And another one is a composition analysis of the ingredients, whether it is going to be a fish meal or rendered animals or insect or whatever. So palatability and feed intake, digestibility, functionality during the process of extrusion technology and the expansion and all absorption and so on. And of course, at the end, growth, gain, FCR, fecal production and production, product quality and so on. For it is 100% all probably we agree that fish meal and oil meet all these criteria, right? Excellent protein and oil sources, but it is certainly a finite or endangered sources. So then let's see what the fish meal history uses. In between 1950 to 1990, it starts with the pig, cattle, and poultry. And in 1990, it starts with an aquaculture. So aquaculture was the, let's say, the dominant consumer of fish meal. But then, right now in 2020, according to IFO, International Fish Meal and Fish Oil Organization, we are much more used fish meal than other industry in terms of pigs and poultry. So as you can see in this graph that we almost 80% fish meal is consumed by the aquafeed industry. So, but another important and interesting thing is that the Ron Hardy, who is the writer of and the editor of the Fish Nutrition book, he was mentioning in 2010 that according to Ron, he postulated that fish meal will not longer be the primary sources in aquafeeds. So what he says is that it will be a special ingredient added to enhance palatability, like an additive, not for protein sources or oil sources, something like that. So it will be an additive in the feeds of aquaculture industry. Is that true? I think it's true because of the fact that between 1975 up to, let's say, 2000, aquafeed version 1 was around like using a fish meal and oil based. In aquafeed version 2 between 2000 and 2015 was related with the law, being mostly replacing fish meal and oil with plant-based oils. And 2015 to up to now, it's changed a lot because we started to use some marine ingredients, byproducts, and some other really important strategic ingredients and functional ingredients. What we have done, concerned research efforts have contributed towards the reduction of fish meal and fish oil in between 2002 and 2010. And right now we really achieved some way. For instance, this is another example which I told you that Hua et al. 2019. And as you can see here that in rainbow trout, there is almost no fish meal. This is an experimental, let's say, result. And in shrimp, it's around 4%. And European sea bass, which is a very carnivore species in Turkey and Europe actually. So it contains only around 15% fish meal, and probably we are going to have less. In terms of other species, with the projection of taken and methane in 2008, what they assume that in 2020, right now, the consumption of fish meal or the contribution of fish meal is going to very low. So in catfish, it will be 0. In carp, in tilapia, it will be around 1 or 2%. As you probably know from your perspective that we achieved this numbers. But when we consider, let's say, a more carnivore species like salmon, trout, and marine species, we really achieve to replace fish meal, for instance, nutritional value, customer acceptance, availability, price or cost, and so on. So, but I will give an in salmon industry. So the salmon industry grows very fast in Europe and all around the world, in Chile and Peru, even in China, I know that. So when the production and total feed use increase, the usage of fish meal and fish oil decrease a lot. So then we have another challenge. We are going to have a less EPA and DHA in the fillet, so which creates a problem for the consumer's perspective. But this is not our issue, but we have to consider this one as well. 1 month ago, there was a webinar, and I took this slide from there. So Margaret from the Norwegian Technical University, she emphasized that the aquaculture, salmon aquaculture industry in Norway, which is a very dominant industry right now in Europe in terms of marine production. And he says that they, in Norway, they use almost 12% fish meal in their diet, and 71% of salmon feed is almost plant-based, or other ingredients is going to come next 10 years. So, but the problem is with the plant-based production is intensified crop production, pressure on land and water resources, and of course feed food competition, which I explained and told before. So by 2030, all fish feed should come from sustainable resources. So then how we are going to be to make this sustainability? So Taken et al. In 2021 has a let's say estimation with respect to production of aqua feed and also fish production. So they think that if the increase will go with this way, let's say a 7.7%, then we are going to need almost, it was, as you remember, 52 million tons. So it will be a 70 million tons. So we need definitely a sustainable resources and of course cheap resources. Oh my God, what happened? I think I have, let me send, hold on a minute. Can you hear me?
Yes, yes, Prof. Please go
Okay,
ahead.
just, I don't know why, but the PowerPoint doesn't want me to follow because I speak too much, I think. Okay, I will go fast. I'm sorry for this technical issue. Okay, I was here, right? Okay, so then when I took some figures from the reports of private companies, so they think that the plant-based diets, it's around 0.5 or 0.8 dollar cents. And if we produce diet with fish and fish byproducts, it is around 1.3 to 1.5. So in Europe, the production cost of diet is around $1. If you produce it less than $1, then you are going to do good business. But when we consider the microorganisms or other alternatives, then the price goes up because of the production cost of these ingredients. So then, let's see, is Fishmeal-free concept, which was with a sharp title, but is it realistic or not? Actually, I took this presentation slide from the presentation from Grengos, this one, and he just put excellent photos. So that's why I just want to share with you. So these are the alternatives that we have in our hand right now, even low or high scale production and so on. But when I read different articles, current literature, where they try to find functional raw materials, these are an excellent articles that I really recommend who is working on alternative ingredients, because these researchers hasn't emphasized that we are going to do it, but we have to do a lot, which I will talk you later. So skyrocketing raw material price, scarcity of quality raw materials, and dwindling global supply chain have put a serious pressure on feed formulators and on nutritionists and on industry. So worldwide, to provide quality feed at sustainable price will be the key issue. So what are looking for functional novel ingredients in aquaculture? First is first, very, very hot topic, develop sustainable ingredient with a low carbon footprint. So that's very, very important, underlined, red marked issue in here and all around the world, I think. And maximize the use of existing ingredients. So there are, of course, different alternatives, but I think in Biomin and Biomar, things that these four sources, plant-based sources, terrestrial marine sources, and rendered byproducts and unconventional sources will be feature of fish meal substitutes. Okay, let's go with small details in each one. I will introduce you some SWOT analysis in these beautiful ingredients. So as Dr. Raji explained, that plant-based protein sources are very, very important, soybean, rapeseed, sunflower, and so on. But we have to consider the let's say a replacement, ratio in the diet, because of the fact that immune response is highly influenced by the nutritional background, and use of plant-based diets is often accomplished by intestinal inflammation and inflammatory cells, as explained in previous researches, that it is very important. So that's why we use these kind of organic acids or other alternative additives to cooperate together with these plant-based product or other ingredients which will be used in the future. In addition to that, their wide range of uses for human feeding, cattle feeding, and biofuel production, which will be let's say, which have been compete our industry. Actually, we compete with our industry because the price is going up and up. So this can create a big problem in the industry, because of these competitors among industry will have problematic concerns in this respect. On the other hand, when we think nutritional quality of alternative ingredients, they are not equal to that of fish meal and oil. So common drawback is low protein content in balanced amino acids, which is explained by Dr. Rajiv, that poorly available phosphorus, and lacking, let's say, in terms of fish oil sources, long chain fatty acids, and so on. So depending on development stage also is very important, and sudden change in physiological stage can affect the fish. So that's why very, very important concerns should be taken into account when we are thinking the plant-based proteins, which we know because we have been replacing them for the last 20 or 25 years. Okay, so one of the issue is the anti-nutritional factors. So there are many anti-nutritional factors, and also plant-based proteins has some mycotoxins, which is very important. Since 2015 to 2022, this is very delicate issue in Europe. So when we have a look the ingredients produced in Asia and Europe, there are many mycotoxins in their let's say composition. And Rue Conchao from Biomin, he explained that in the finished diets that we use in the grow out period, almost 25% of finished diet has more than one mycotoxins in it. So this can be another problem for inflammatory perspective and health perspective of fish. So let's have a look of the plant-based protein sources SWOT analysis. So production method is settled and established, and most are byproducts of human foods are working very well. The weakness part is processing methods and genetic technologies. Sorry, the opportunities is going to processing methods, which we use the heat and pressures in order to increase these inhibitors in the plant-based protein sources, and genetic technologies, which creates a great opportunity. In Canada and UK, for instance, they produce a plant which contains LC-PUFA, long-chain fatty acids, EPA and DHA. So it contains almost 20-25% EPA and DHA on itself, on the leaves of the plant. So that will be a genetic technology. However, we have some weaknesses. We need some long, let's say, LCA wise, because from the process till the end, we have to use these the LCA calculations and nutritional characteristics, like in balancing digestibility, amino acid composition, especially lysine-arginine ratio and lysine-methionine and lysine and leucine ratio in the plant protein sources. And of course, the trade are climate change, water usage and land usage, and competition with the population growth and demands for the human in this respect. When we'll come to terrestrial or marine sources, I just want to focus on single cell proteins, which are very, very hot topic right now in Europe. And there are many European Union calls related with these single cell proteins. And as we know that they increase the digestibility and has a nutritional value compared with the fish meal, and an absorption of EPA and DHA is really high. So this is very important opportunities from these three guys in here: bacteria, fungi, and macroalgae. But it could easily be let's say researchers think that in 1 or 2 years time, we see affordable macroalgae with good DHA and EPA levels coming in the market. And we do know that in Europe, the production of these microalgae and bacteria is going to be high. But production volume costs are still major constraints for a wider use in aquafeed industry. So that's why competition with the human and biofuel production is going to another challenging part. And the variable of amino acid composition is also, let's say, critic points that we have to take into account compared to fish meal. So it is variable crude protein levels in their composition, and as I said, quite acceptable essential amino acid, which can be increased by using, let's say, cell walls and other things. But I will not go in deep since I don't have enough time for that. So microbial ingredients by using natural gases has promise in aquafeed, high nutritional content, sustainability terms, they are very important, and there is no any competition with human food, so that's another important component for them, and they can be produced year-round. Not on, it's not, let's say, affected by the climate change or seasonal changes and so on. And one of the alternative guy in here, methane utilized bacteria, encapsulatus, which contains around 70% protein, very good balanced amino acid, and 10% nucleic acid, which is very important for immunology and growth performance of animal. And it contains bioactive compounds like methanobactane and MAP-E or peptinoglucans. So this it can be another candidate which has been working in Norway for the last, let's say, 5 years, but also it contains a very good production efficiency. Not only bioactive compounds, but also production efficiency is very important when we consider these kind of microbial ingredients in Aquafeed formula. So, of course, we have some, let's say, strong opportunities. As I told you, digestibility, high nutritional value, some progress in mass production achieved right now. And in China, I think this week or next week, one factory is going to produce this bacteria that I told you in my previous slide. So they plan to produce around 100,000 tons within a year. So this is very promising, actually, and excited news that I heard just 2 weeks ago. And they can be a functional ingredients, not only replacing fish oil, but also a functional ingredients to boost immune parameters of the animals. And heterotrophic single cell proteins are having opportunities for the next generation functional feed, as we think that they are going to be. And also the weakness part is they have EPA and DHA content differs, as I said, and cell wall integrity and wall type can be a weakness part. But as we know that in Netherlands, in Wageningen University, 2 new Ph.D. released that they overcome this cell integrity and wall type problem. So they have some excellent results, which I'm not going to present you, but you can easily find in the literature. But this is already solved. And the trade is the mass production and productivity need improve, and the price per ton has a long way to go. So that's another trait. I will just try to be fast because I think I have run out of my time. But rendered byproducts are very, very unique, and it has been used for the, let's say, 6 years or so in Europe because of the fact that they are of high quality, economically affordable, and environmental sustainable. So the biosecurity, circularity, and or LCA terms, we have to consider these rendered byproducts. Well, actually, as you know, that in Europe or in many other countries in the world, in 2013, the EU lifted the complete ban on pups used in animal feeds, but you can use these byproducts in different animals. But the high standards must be employed regarding the production, storage, processing, and distribution and tracing of such products will have been very important, and then it can be used in animal nutrition in the industry right now. So trimmings are fantastic resources, and IFO and University of Stirling publish an overview with this respect in 2016, and the production of raw material from the fish byproduct is going to be 20 million tons. So when we consider now, IFO says that 29% of world fish meal production already comes from the byproducts. It is around almost 4 million tons. So this is very unique, actually, numbers. And what they expect is that in 2025, extra 9 million tons fish meal is going to come in the industry by using the aquafeed industry. So this is very, very unique. And 1.5 million tons oil is going to come within let's say, within 3 or 4 years in the industry. So of course, there are some, let's say, strengths and opportunities, but also weakness and traits in terms of rendered byproducts. increased digestibility, high nutritional value and price, excellent. And growing production and oil comes from the byproducts are very opportune, having an opportunity for the industry. And the weakness is regulation and quality standards, which can be changed during the season and during the harvest time. And standard production is not ready yet. And trait is high variability in the composition and protein quality, like macroalgae or single cell proteins, as I explained to you before. This is very, very hot topic, I think in Europe, but in Asia, as we know that you use insect proteins and oils for a long time, but in Europe, the people are very excited for these unconventional resources. This is a very new results of released by the Rabobank. And we know that they are very good candidates which convert low value organic materials to the ingredient, and low environmental impact, high nutritional value, and there are some legislations in Europe to be able to use these products in the industry. As you can see here, that aquaculture industry can use these byproducts as protein, fat, and even live larvae in the under the pertinent conditions according to legislations certified by the EU, but you cannot use whole or dried or frozen ingredients. So you have to produce some protein and fats from these ingredients. And the insect companies that have received the largest investment and stand out for their business for the last, let's say, 5 years. So as you can see here that $430 million has already invested in this industry. And we do know that the Thai Union CP start to produce and start to invest some money on insect production, because it is quite a very important, I think, resources for the animal production, not only for fish, but also for pet food and animal nutrition. By 2030, the Rabobank estimated global market potential is going to be 500,000 tons. You can see here that then the maturity phase is going to finish, and the price of per ton is going to be around 1,500 or 2,500 euros, which is quite equal with the fish meal and oil. So in this respect, 200,000 tons is going to allocate to aquaculture in 2030. Still, it's low and high cost compared to fish meal, and legislation and food safety is very big concerns when you start to produce these ingredients. So, but the projection is 500,000 tons within, let's say, 25 years or so. As I said, in this article, they criticize and talks about what are the main positive and negative things. But in the case of, let's say, insects, it is protein. It contains a high protein, environmental sustainable, consumer expectant. Actually, still there are some concerns about consumers' expectance, but when it comes with the feasibility and cost and environmental sustainability, then the consumers change their altitude on the diet or on the fish produced by the diet containing insect meals and so on. So these are having the weakness and traits for these unconventional sources when we consider the SWOT analysis. So, okay. Fish meal is just an excellent sources of high digestibility, as we know, high palatability, dietary protein with an excellent amino acid composition. But fish meal not be a longer protein sources, but it will be, as Ron Hardy explained, that it will be like a substitute ingredients compared to other ingredients or compared to the last 10 or 20 years production period. So fish meal can be replaced up to 0%, but it will cost. If we are going to consider algae and others, perfect, but it will cost us for now. But within 20 or 10 years time, the cost is going to decrease, and marginally, I think the replacement ratio is going to increase in due course of time. Right now, we can say that no fish, no shrimp needs any fish meal. However, they need in their diet or in their formula essential amino acids, correct protein and energy ratio, less per unit of feed, but better use is going to be considered. So my last slide, towards to fish meal-free diets. I think we have done enough, but in terms of product quality and production perspective, we have to do a lot. I think we have done enough, as I said, concerning the above mentioned items. However, a lot remains to be done in the future. Thank you very much.
Thanks so much, Dr. Prof. Tufan, for your presentation. I think you cover a lot of knowledge on replacement of fish meal and fish oil with the functional novel ingredient. I think especially for the insect meal, yes, it's very hot. Even in China, hot topic. I think plenty of researchers, they are doing this kind of a project and the research. Yeah, hopefully in future we can produce stable and a big quantity. Okay, let's move to, I think, one question for you, Prof. Tufan. So you have mentioned the insect meal in your presentation. So could you share some application progress of insect protein in aquaculture in Turkey or Middle East? This is a basic information, I think. What's the bottleneck for the insect protein? And what's the solutions to overcome such kinds of bottleneck? Yeah, if I'm not wrong, I think so far we can, all over the world, the total production is around 10,000 tons, right? About that for the insect meal. So please.
Thank you for the question. It's an actually excellent question in terms of when I do this kind of presentation, the first question comes from the audience is going to relate it with insect meals. That's why, as you said, Dr. Bai, that it is a very hot topic. Right now in Turkey, no one uses insect meals because of the price, which is around 2.6 dollars per kilo. It's so in Turkey we use our fish meal, and we harvest some fish meal from the Africa. So we have some factories over there. So that's why the price of fish meal is quite, let's say, less cheaper than insect meal. But it will come. When I talk with the people in Europe, it is definitely be the future of a replacer of fish meal. As far as I know, that in salmon industry, they started to use because they cannot get this fish meal just like us, because we produce our fish meal in Turkey, as I said. So that's why they paid almost the same money for fish meal and insect meal. So they choose this way because of the fact that in Europe they must follow the green deals. So the green deals force you to use this kind of sustainable resources instead of using a fish meal and oil which is going to be harvested from the ocean. So that's one of the things. As Turkey, we are going to be as an associated country, we probably is going to follow the green deals in 5 years' time. So we have some commitments. Our producers have some commitments to European Union in order to export some fish. So we will replace some insect in the diet. In the Middle East, I'm not sure, maybe in Egypt, probably they started to use, but the production cost is again quite high. I'm not sure in Middle East, the people, let's say in Turkey case, will not use for now insect meals. But there are some applications to decrease the price of production insect meal. Then within 5 or 10 years, we will use them as an ingredient.
Okay, okay, that's great. Okay, any other questions? So I think that's all for the Prof. Tufan. Thanks for sharing and a very knowledgeable presentation.
Thank you very much.
I did learn a lot from your presentation. Hopefully in future we can face-to-face discuss or have a talk about this again. Okay, thanks again.
Thank you.
Okay, hi, bye-bye, thanks. Okay, so let's move our last speakers, but last not the least. She is a professor from Kazakhstan University, Thailand, Department of Aquaculture. During her Ph.D., she conducted her research in Tokyo University of Fisheries. She is expert in aquatic, animal nutrition, feed technology, aquaculture system, waste management, like the wastewater maybe, and the shrimp culture in Asia. Her specific focus is research on feed additive and fish meal replacement. Fish meal replacement again in Aquafeed for sustainability of aquaculture business. She has published plenty of papers. Please welcome Professor Orapint Jintasataporn. Doctor, Prof. Orapint, now it's your time.
Okay. Thank you, Dr. Bai. I don't know
Welcome.
how to. Okay, I can open it. Yeah.
Okay, please.
Dr. Bai. Good afternoon, everybody. I try to share the slide, but I'm not sure how to. I'm not sure now I share it or not. Cannot start video. I don't know. Oh, okay. Yeah.
So you try the red corner.
Yeah, thank you.
Okay.
For the topic that I would like to present today is the lipid digestion and the hepatopancrease help for promoting shrimp growth performance. So because of the aquaculture business grow very fast and grow continuously more than 10 years at the rate very high, roughly around 10%. And the aquafeed market also grow very fast. Yeah. From 2020 to 2020 to 2025, the value of the aquafeed, I think, is increased more than 20 billion. And when we talk about the aquafeed, yeah, we have 3 parts of the feed that we concern. The first one is the protein source. The second one is the energy source, and the third one is the micro ingredient. So today the problem of the aquafeed is the raw material price increase very fast and very high, and we the business expect this increasing, the price increasing will continue as more than 2 or 3 years due to many reasons, and also the logistic price also increase. That's the reason why we go to, or we try to reduce the feed cost and also try to improve the efficacy of the raw material in the feed. Yeah. So as we talk about the raw material today, most of the researchers try to reduce the level of the fish meal because of the price is high, and increase using the plant protein material because of it's cheaper. But when you using the plant material, we face a lot of problems on the anti-nutritional factor, especially on the fiber, phytate, or some toxic ingredient, or some what you call anti-nutrition or like the protein inhibitor, yeah, in the material. Not only the protein source, the lipid also face the problem. The price of the fish oil increased, and also the price of the soybean oil also increased a lot, as you see in the graph. And this one, yeah, you can see the price of the fish oil increase, and also the price of the soybean oil also increased. But anyway, the price of the fish oil still higher than the soybean oil. That's the reason why some feed mill, they try to replace some part of the fish oil by using the soybean oil or the vegetable oil. But the composition of the soybean oil or vegetable oil is big different from the fish oil. You can see in tuna oil. Tuna oil composed of the long-chain fatty acid, omega-3 long-chain fatty acid like EPA and DHA at very high level. Sardine oil also the same. Both of these fish oil contain high omega-3 long-chain fatty acid, EPA and DHA. Not only the long-chain omega-3 fatty acid, inside the fish oil, it also composed of the omega-3 PUFA like linolenic, yeah, and also a little omega-6 like linolenic. Compared to the vegetable oil like soybean oil, corn oil, we can see no long-chain fatty acid, EPA or DHA, but it contains high omega-6, the linolenic, and also a little omega-3 linolenic, yeah. Soybean oil seems to have more linolenic than another vegetable oil. Palm oil, just only a few omega-6 and omega-3. Most is the omega-9. When you use the palm oil, when you use the vegetable oil compared to the fish oil, one thing that we can notice is the lipid digestibility. Lipid digestibility of vegetable oil is a little bit low, like this one, yeah, come from the palm oil, yeah, and also some from the soybean oil or the corn oil. Yeah. The digestibility is lower than the fish oil. Yeah, fish oil mostly is the long chain fatty acid in this term. Digestibility of lipid more than 90%, yeah, more than 90%. So when you use vegetable oil at the high infusion rate, yeah, and reduce the fish oil, we will face the problem of lipid digestibility, and the animal will get less energy for their growth performance. For the nutrient digestibility and absorption, we have some point that we can notice. The aquatic species like fish and shrimp, they have high nutrient digestion and absorption, especially on the protein and lipid, due to the wide range of pH and temperature for enzyme activity. This one very important because of the enzyme. Most of the enzyme, they have their specific activity on pH and temperature. If the enzyme have the wide range, it means that when the climate change or when the feed composition or feed quality change, the enzyme still active and can digest more nutrient and provide nutrient for the animal body. And also because of the shrimp and fish diet, especially the carnivorous fish and shrimp, inside the feed is composed of high lipid. This high lipid will retard another nutrient digestibility. Because of the lipid digestion, the enzyme work on the aqueous condition. When they digest the lipid, they need to lysolable the lipid or compose the lipid in the water phase. That's the reason why they need some emulsifier. Inside the fish or the shrimp body, they have the emulsifier in terms of the bile acid. Fish very clear. For the shrimp, we are not we are not clear on the bile acid synthesis by itself, but they have some requirement on the phospholipid and the cholesterol that will help to digestion the lipid from the diet. So the bile acid very important for improve the digestibility, the lipid digestibility inside the animal body. And bile acid work as the emulsifier to promote the enzyme activity during the lipid digestion in the digestive tract of the animal. So how to improve the nutrient digestion and the absorption? One technique or one research that we found and the efficacy is really good is supplement the exogenous enzyme to promote the nutrient digestion, like supplement lipase, protease, carbohydrates, or phytase. Or we can add some additive for enhance the enzyme activity, like using the emulsifier. Many emulsifier we can add in the diet, like the bile acid, the lecithin or lysolecithin, and some stabilizer like the monoglyceride and diglyceride incorporate with a little acidifier will stabilize the condition of emulsification and can improve the lipase enzyme activity for digest the lipid, including can promote the digestion of another nutrient like the protease or the amylase activity. For the acidifier that we can apply or we can use in the diet to promote the digestion, we can use citric acid or propionic acid, including supplement some mineral like zinc, copper, manganese, magnesium, and iron for to be the cofactor to promote the enzyme activity or enhance the enzyme activity. Another part that we can improve the nutrient digestion, we can focus on the gut health and the epithelial permeability by supplement some probiotic to control the gut condition or prebiotic to promote the bacteria in the digestive tract and improve the gut health, or use some organic acid for to be the energy support the epithelial cell activity, like supplement the butyric acid, the malic acid, succinic acid, lactic acid, or group of the medium chain triglyceride like the lauric acid. Because of this group, when the animal consume feed with the medium chain triglyceride, medium chain triglyceride have some function to control the bacteria inside the digestive tract and also can absorb very fast for to be the energy for enterocyte cell or for the epithelial cell that will promote the villi function. And also reduce the gut inflammation. Some herb extract like the garlic, turmeric, or the oregano oil. We found that it can improve the digestibility of nutrient in the digestive tract. Because of this herb extract or essential oil, they have some anti-inflammation function and can provide can be the antioxidant for promote the enterocyte cell function. Not only the herb extract, some mineral also can reduce the inflammation, especially the zinc. Zinc have some pathway to inhibit the express of the NF-kappa B, so it can reduce the inflammation of the animal inside the digestive tract, and also reduce the mycotoxin. By using the mycotoxin binder, we can reduce the gut inflammation. Probiotic also one material that we can use to reduce gut inflammation via the secretion of many metabolites from the probiotic. The other one that technique we can can improve the nutrient digestion is the grinding techniques to grind the raw material to the small particle, increase the surface of the material. After that, the animal can digest very easy and release more nutrients. Or cooking cooking the raw material by using the extruder or pellet machine, because of when the raw material cook, especially for the starch, cooked starch compared with the raw starch, the digestibility increase more than 20%. Yeah, or fermentation technique to cut the nutrient to the small pieces, especially the protein to be the protein hydrolysis. Yeah, that will easy for digest and absorb. Yeah. So about the bile acid, because of lipid, generally in normal condition, aquatic animal can digest lipid very well, but when you change the raw material, when you change the protein from the animal protein to the plant material, they have some anti-nutritional factor. And also when change the fish oil to the vegetable oil, yes, the composition of the fatty acid or the triglyceride will affect on the lipid digestibility. So adding some bile acid both for the fish and the shrimp. Yeah, the fish very clear bile acid, very useful to be the emulsifier and promote the lipid digestion. For the shrimp, inside the bile acid, it have the bile salt and also they have some cholesterol. You can see the level of cholesterol in the bile acid a little bit high, and have the fatty acid, including the some lecithin. So this bile acid, when we add in the feed, it will promote the digestion of the lipid and release more fatty acid for absorb to be utilized in the animal body. So for synthesis, the bile acid, we have some steroid to be the predominate in the bile in the mammal and other vertebrate like the fish. And this steroid acid will divert to the bile acid in the liver, and after that they will convert to the bile salts by using taurine and glycine. And this bile salt, including the bile acid, is will accumulate in the gallbladder of the animal and released when the animal want to utilize the nutrient. Yeah. Bile acid synthesis in the hepatic organ. This is very important because of the health, the hepatic function or the hepatic health, very important for control the metabolism of the animal. So for conclusion, we can say that the bile acid made in the liver and stored in the gallbladder. After that, this bile acid will help the digestion and absorption by to be the emulsifier in the lipid digestion process and make the lipid soluble in the water, easy for the lipase to work. Yeah. So for the composition of the bile acid, it composed of bile salt, phospholipid, cholesterol, and the bile pigment that we call bilirubin. Yeah. This picture show you the the activity of the bile acid. Bile acid have two parts. One part is the hydrophobic and the other part is the hydrophilic. So when the animal want to use this bile acid as the emulsifier, the bile acid will compose or will bound with the triglyceride in the diet. After that, they will use this bile acid, yeah, to cover the triglyceride, and the area between one bile acid and the other bile acid is the area that the pancreatic lipase will come to digest this triglyceride, because of after the function of the bile acid, the lipid already mixed or already dissolved in the water, easy for the lipase to work and cut the bond and add the water inside the molecule. Yeah, this molecule, this triglyceride cover with the bile acid. We call this molecule micelle. For the micelle after lipase function, they will release the fatty acid and the monoglyceride or the glycerol. This molecule will absorb, pass through the enterocyte cell and compose to be the new triglyceride and carry from the blood circulation to the in terms of the chylomicron. Chylomicron in the animal, in human, or in the livestock and fish. Yeah, we have the form that we like of the lipoprotein that we call the HDL. HDL after carry the cholesterol and some triglyceride, including the vitamin, the fat soluble vitamin and the carotenoid. They will carry via the blood circulation to the liver, and then after to the liver, they will release all the nutrient inside, yeah, and provide the nutrient, the lipid, the vitamin and carotenoid for the liver cell. The liver cell will have the metabolism to pass this nutrient to the cell that need the nutrient for their function. For conclusion, the activity or the role of the bile acid, yeah, at the liver, they synthesis at the liver and store at the liver and release for improve the lipid digestibility. So bile acid have the metabolism relate to the glucose production, relate to the cholesterol and triglyceride metabolism. This one happen on the liver area. So the nutrient in the blood circulation, after they have the metabolism via the bile acid function, they have some trigger to the brain. Yeah, like inside the brain, they have the appetite area and also have the satiety area to control the feed intake. Yeah, when the level of the glucose low or the energy low or the protein inside the blood circulation low, it will trigger the appetite condition from the brain to consume, to trigger the animal to consume the feed. Yeah, after the meal, yeah, they have the food in the intestine, in the stomach and intestine. This one will trigger the secretion of the bile acid to the digestive tract and promote the lipid digestion. Not only promote the lipid digestion, they also trigger or enhance the secretion of the insulin, which relate to pass or which to absorb the sugar to the cell. Yeah, this one is the insulin. Yeah, this is the one part. Another part is bile acid after the meal, yeah, and have the nutrient from the blood circulation. It will trigger the muscle, yeah, the skeleton muscle, but via the insulin function to absorb the glucose or another energy to the muscle cell. Yeah, and this one will promote the muscle yield function. And also, yeah, from the bile acid function, they also trigger the insulin secretion and utilize the energy for the lipid carry from the digestive tract to the liver. This lipid will deposit in the adipocyte cell or utilize depend on the energy that the animal get at that time. And also this bile acid will relate to the decrease of the cytokine release. This one will relate to the immunity of the animal by reduce the inflammation inside the digestive tract. This is the function of the bile acid, yeah, that we found in the animal body. In shrimp, maybe the function not too clear like this, but most of the most of the function quite similar. We can imply via the result of the growth performance and some immunity. So how to boost or how to push the bile acid activity? We can add some more the choline, because of choline assists with the fat digestion. Choline and glycine boost the bile acid production, change the bile acid to be the bile salts, and also pancreatic lipase can break down the fat as the digestive enzyme. Exogenous bile acid also provide the essential bile salt to be the emulsifier, promote the lipid digestion, and beetroot and betaine from the beetroot also help to provide or to avoid the gallstone accumulation of the calcium inside the gallbladder. Yeah. So for the shrimp inside the hepatopancreas of shrimp, we have some accumulation of lipid, and this one is the sign to inform the health of the hepatopancreas, because of if the shrimp healthy, it will accumulate more fat deposit in the hepatopancreas to be the energy during the molting period. Incorporate with the glycogen. Glycogen release the energy faster than fat. That's the reason why in the hepatopancreas they need to accumulate both glycogen and lipid. Yeah, and they have the small fat droplet inside the hepatopancreas tissue. So for the hepatopancreas tissue, they have the cell, 4 types of cell inside the hepatopancreas tissue. The first one is E cell. E cell is the end of the distal area. This one is the is the cell for synthesis or for change to another 3 cell. Yeah, it's the like the nuclear body to give light to other 3 cell type of the digestive gland. So E cell can be the F cell or B cell or the R cell. Yeah, depend on the differentiate of that cell. The other 3 cells that really important inside the shrimp hepatopancreas is the F cell. This one is the F cell. So from the histopathology, you can see the F cell like this. Yeah, at this area. The F cell is responsible for the protein synthesis and storage the mineral. The B cell is the last cell. B cell is the last cell. Yeah. This last cell is synthesis, the digestive enzyme, and also responsible for the nutrient accumulation. Yeah. Intracellular digestion transport the digestive material to another cell and also pass through the intestine after absorption. The other one is the R cell. R cell is the triangle cell composite like this. Yeah, it's the multi-vacuole cell occur throughout the hepatopancreas and have absorptive and lipid and glycogen accumulation store for using during the molting period, including accumulate some mineral like calcium, magnesium, phosphorus, sulfur, and other mineral for support the molting. Yeah, this one is very important. So hepatopancreas of the shrimp is very important because of they accumulate everything inside and utilize it for during the molting cycle and make the shell harder to protect itself from the prey or from another animal. And also the hepatopancreas is function like the intestine in the livestock or in the human. Inside this hepatopancreas, they have the digestion and also have the absorption. If the hepatopancreas unhealthy, it means that this animal face the problem, yeah, and on the state of risk to get the disease and malnutrition. So to evaluate the quality of the hepatopancreas, yeah, we have 2 techniques. The first one is the wet mouth. wet mouth like this. Yeah, we collect the tissue and cover with the cover slip and observe under the microscope. Yeah, at magnifies 4 or 10. Yeah. So the hepatopancreas, we can see like the finger is round, yeah, and the tip end close. Yeah, inside they accumulate a lot of oil droplet. Yeah, to show the healthy hepatopancreas. More oil droplet is more healthy hepatopancreas. Round shape is good. If they have some constrict like this, yeah, it's not so good. Yeah, and during this time, one parasite, someone called parasite, someone called just only pathogen, yeah, that infect in the shrimp everywhere in the world is the EHP or Mycosporidia. We call it hepatocytosine. This one, the size is very small, and in fact, in every part of the hepatopancreas cell, yeah, they can infect R cell, infect B cell, infect in the F cell, yeah. So for evaluate or for ranking the quality of the hepatopancreas health, we have the healthy completely hepatopancreas, the healthy hepatopancreas, the hepatopancreas cell is loud like this, yeah, and have the oil droplet accumulate a lot. For the unhealthy one, you can see some constriction like this, yeah, some defect, yeah, and also sometimes it's swollen, yeah, and sometimes they lose their epithelial cell, epithelial tissue, yeah, inside we call it ATM, yeah. This one show the healthy hepatopancreas, yeah, cell, yeah. Okay. And this one unhealthy, they have some constriction. You can see constriction like this, yeah, but they still have oil droplet deposit, but not too much compare with this one, yeah. And also we need to grade, yeah. This one is the low oil deposit, yeah. You can see some space on the hepatopancreas, and this one is the medium oil deposit, yeah, lipid deposit, yeah. This one is high oil deposit, yeah. Although they have some constriction, but oil deposit at the high level, yeah. And this one we call it medium hepatopancreas. Yeah, melanization of the shrimp. Yeah, they have some. In case of the shrimp have some infection by the bacteria or the parasite, yeah, they will release the high oxidizing agent like phenol oxidase to control the pathogen, yeah. After that, this phenol oxidase will change to be the melanin, yeah, and they will accumulate some melanin in the hepatic cell, in the hepatopancreas cell. That's the reason why we see some melanization in shrimp hepatopancreas. Yeah. And also the other side of unhealthy shrimp we call aggregated transformed microvilli or ATM. ATM is the epithelial cell of the microvilli and loose. Yeah. Someone said in case of the animal unhealthy, we will see a lot of ATM or the epithelial cell loose. Yeah. But in some case, the healthy shrimp also found the loss of the epithelial cell of hepatopancreas or epithelial cell of microvilli because of in general, like the animal or the livestock body, the digestive tract, epithelial cell of digestive tract will lose and synthesis every day. Yeah, it depends on the condition or it depends on the quality of the feed and material passed through the digestive tract will irritate. And sometimes because of they have high activity, yeah, this epithelial cell die and synthesis the new one. So, but it's just some sign that we can observe or we can see. Yeah. Not only this parameter, we need to see or we need to evaluate the shape or conformation of the hepatopancreas is loud or constrict, or they have the accumulate of the lipid, they have the melanization or not, have ATM or not, have some infection of the EHP or not. Yeah. And also for the intestine, we can see the information of the intestine that we call enteritis by see the sign of the information. Yeah, like this. The color is a little bit red. Just inform the information. In the normal cell, the pigment is shrink and compact. Yeah, and we can see a little bit dark spot than the unhealthy intestine or enteritis. Yeah. Okay. In case of the bacteria infection in the hepatopancreas, yeah, we can see some sign of the inflammation. We can see a little bit red or pink color, yeah, spread or dispersed inside the hepatopancreas like this. Yeah. And you can see this one is some sign of the inflammation. And also after infect a few times, this one will change to the melanization. Yeah, this one show the bacteria infection. So, but by this infection, we cannot say this one come from the pathogen or come from the beneficial bacteria. A lot overdose in the system, in the culture system. Yeah, and a little bit ATM. Yeah. So for the hepatopancreas, it's very important because of hepatopancreas is the organ that animal use to synthesize and secrete the digestive enzyme for digestion, absorb and digest the material, transport and store the nutrient to the muscle, to the gonad and other tissue during the growth and reproductive stage. Help in the metabolism of lipid and carbohydrate. Be the storehouse for the large amount of the energy. Hepatopancreas is the area to keep or to store the energy during shrimp molting. For supply energy for molting, starvation or reproduction, synthesis of the vitellogenin and the steroid hormone act as the site of the delivery of the mineral or the organic product. Yeah, can synthesize everything like the big factory, synthesis everything and provide the substance or material to another cell. Yeah. So if hepatopancreas have some problem, unhealthy, it means that the function, they cannot synthesize the substance provide to another cell. So the shrimp will on the state of risk on the malnutrition and also risk on the disease, infection. Okay. And the effect of the hepatopancreas health on the shrimp growth performance. If the animal face the problem on hepatopancreas health, we will found the retard on growth, the size-wise variation, demonstrate the ATM is aggregated transform microvilli. Sometimes we can see the milky body, the back scar present on the shrimp scale on the shrimp shell in low salinity culture, or sometimes we can see the soft shell because of they cannot accumulate more mineral and also cannot utilize mineral to accumulate on the new shell, make the shell hard, more susceptible to the disease, and also exhibit the white fishes syndrome. The high FCR and the low production is the sign that we can see. For the try on the effect of bile acid on enhanced hepatopancreas health in white shrimp, we apply the bile acid in the diet. So we have two groups of the diet. The first one is the positive control. Positive control composed of the fish meal 15% and 2% of fish oil. Yeah. For the negative control, we reduce fish oil to 1%. Yeah. This treatment did not add any bile acid. For the treatment 3, negative control, with fish oil 1%. We add bile acid 0.015%. The next treatment add bile acid 0.013%. 0.03%. And the last one is negative control, add the bile acid 0.45%. After we culture it for 8 weeks, we can see that the negative control without adding bile acid, the size of the shrimp a little bit small. When we add more bile acid, the size increase. Yeah. And for the negative control with the bile acid 0.45%, yeah, the size compared to the positive control quite close together, you can see. Because of inside the bile acid, it contains cholesterol, fatty acid, in terms of phospholipid and also lecithin. So this one very important for the shrimp growth performance because of the growth hormone of shrimp is the steroid hormone, not be the protein hormone different from the fish. So for the weight of the shrimp after fed the bile acid, you can see, yeah, the weight increase when in negative control group when you add more bile acid, yeah, like this, yeah, and a little bit close to or a little bit higher than the positive control in the high level of bile acid. For the survival rate also improve when we add more bile acid in negative control diet compared to the positive control, but not as good as positive control, a little bit lower, but not big difference. Because of inside the fish meal, not only the fatty acid, yeah, omega-3 long-chain fatty acid is also composed of another material and also have the cholesterol inside the fish oil and phospholipid inside the fish oil. Yeah, that's the reason why the survival rate a little bit lower than the positive control, but not big difference and maybe not significant at this level. For the feed conversion ratio, you can see when you add more the bile acid can improve the feed conversion ratio by reduce FCR. And the important thing for the hepatic or hepatopancreas health, you can see from the color. You can see from the color because of if hepatopancreas health is good, so it means that they can synthesize a lot of material and also can keep the nutrient, provide the nutrient to accumulate at the place that it should accumulate. So the color mainly come from the carotenoid. So carotenoid can be the good antioxidant inside animal body. If the hepatopancreas not healthy, the animal will use carotenoid to protect themselves from many reasons, from the rancidity of the cell membrane, yeah, protect the lipid, yeah, accumulate in the hepatopancreas for to be the energy during the molting cycle. So the clear color of the shrimp, the cooked shrimp, inform the healthy of the hepatopancreas. So you can see the positive control is more darker than the negative control. Yeah. And when you add bile acid, it's not too much clear. It improve, but you can see the improve on the size, yeah, and the color. So after we apply the bile acid to the shrimp, we collect the wet mouth to see wet mouth of the hepatopancreas to compare the hepatopancreas between treatments. Yeah, this one is multiplied by 4, 10, 40, and 100. We can see the accumulation of the fat deposit. Yeah, hepatopancreas health, we give the score. The score we give like previous we inform. We have the low, we have the medium, and we have the high. Yeah. Hepatopancreas health. The score 1 is healthy. Score 2 is the unhealthy. Yeah, so you can see that hepatopancreas health in positive control get the score 1 and have this accumulation around 79%. Compare with negative control. Negative control, the hepatopancreas health is 1.5 because of they have some constriction that you can see from the picture. And also the fat deposit is a little bit low, roughly around 66%. For the third treatment with bile acid, negative control with bile acid, 0.15%. The hepatic pancreas score around 1.4 because of they still have some constriction. Yeah, you can see from this area. Yeah, and lipid accumulation roughly around 76%. Yeah, when you add more bile acid to 0.03%, you can see the healthy hepatopancreas. Yeah, the score is 1.2. Yeah, and accumulate more lipid inside the hepatopancreas. The score is 76. For treatment 5, treatment 5, we add the fish oil to 2% and use the soybean meal at the high inclusion rate, roughly around 40% compared with the normal negative control and positive control soybean meal 32. This one want to show the effects of the soybean meal, high level of soybean meal with have the high anti-nutritional factor. So when we increase the soybean meal and we add more fish oil and also add more some bile acid to 0.45%. 0.045, sorry, 0.045. Yeah, you can see the fat deposition a little bit lower than treatment 2, treatment 3, and treatment 4 because of some effect of the anti-nutritional factor effect on the utilization of energy. Yeah, although the bile acid can increase the lipid digestion, but it's not enough for the energy utilization to compensate the anti-nutritional condition. Okay, and for the intestine, yeah, intestine, we also collect the intestine and see the wet mouth and ranking the score of the enteritis. 1 is healthy, 2 is unhealthy. Yeah, so the lower score we can find with the diet that we add more that we add more bile acid. This one. So this one show you not big difference on the intestine condition in the positive control and negative control. But when you add more bile acid, you will get the good result on the or the intestine to reduce the enteritis. So for conclusion, we can say that supplemental bile acid in fish oil reduction diet with the white shrimp production at the level of 0.03 to 0.045% can enhance the hepatopancrease health, then promote the shrimp growth performance, improve feed conversion ratio, and survival rate. And thank you for your attention.
Thanks so much to Professor Orapint. Thanks for your insightful and informative presentation. It's very useful. We do learn a lot, especially for the standard. This is my first time. I see the standard for judging the healthy of the shrimp through hepatopancrease health index, fat deposition index, and enteritis index, and also the color of the shrimp. I think for the farmers, this is the best way. They just use the microscope. They just use the water for the shrimp. Then they can tell whether the shrimp healthy or unhealthy. This is pretty good. Also for me, this is the first time. And also you share how bile acid works in animal body. Give a lot of photos, the data, like the group, also the FCR. I think this is really pretty useful. And I think we have 5 questions. Let us answer one by one. Okay. Yeah. The first, let me check. The first, what do you think about the efficiency between lysolactin and the bile salt in terms of the emulsification in fish and shrimp, which is the question 1 in Q&A box. What are the strengths and the weakness of these 2 different emulsifiers for biological perspective in shrimp and fish? I think before you answer, I have clarified one thing. Actually, the bile acid based on Dr. Zhang, based on Professor Xue Min, and also Dr. Prof. Orapint, bile acid not only the emulsifier, bile acid can promote the digestion through the increase the activity of lipids, also promote the absorption. They can form the mast cells very tiny and promote absorption too. Okay, so Prof. Orapint, please.
Yeah. Okay, so we can say like this. The bile acid is synthesis from the animal body. Yeah, so we harvest the bile acid from the animal and then we dry it and add it in the feed. So the composition of the bile, like the bile salt, the cholesterol, phospholipid, especially the lecithin inside the bile acid, is come with this bile acid, and it will promote. One part is the emulsifier, but they also have another function, not only to be the emulsifier. When you compare the emulsification property, maybe it cannot as good as the lysolecithin in many parameters of the emulsifier property, but they have another function like trigger the insulin, and also they have the anti-inflammation property, yeah, and also can promote the accumulation of the lipid in the cell and promote the muscle, the protein synthesis inside the muscle cell. That's the reason why bile acid can promote the shrimp growth performance. Yeah. And improve the hepatopancrease function. For the lysolecithin and lecithin, as we know, is the good emulsifier. Lysolecithin more effective because of we cut off one fatty acid to make the molecule smaller than lecithin, and the emulsifier property is high. But this emulsifier property is need to come with another material like some stabilizer, the monoglyceride or the short chain or medium chain triglyceride, including a little bit acid condition to stabilize the emulsification condition for lipase function. Yeah. But
Okay,
they don't have another property for promote the shrimp health or the animal health. Yeah.
Okay. Thanks for your answer. And I think the second question you have given the summary already. Bile acid can promote the growth, lower the FCR and protect the hepatopancrease. Let's move to question 3. Also, maybe you gave the answer already. Can too much bile acid cause a burden to the shrimp health? That means if we add too much in the shrimp feed, will cause a burden or not?
We are not sure, but as the level we conduct, we still not see any problem on the performance. Yeah.
So far, I saw your research, which is I think 450 grams per ton. The shrimp grew well, and also the interiors control the interiors is 1. It's very good, best group. Okay, that means maybe bile acid can control or prevent the interiors, right?
Yes.
Okay. Then other than fat, any additional? The question, other than fat emulsification, any additional benefit for? I think your answer already. This one. Let's move to the third question. What do you think about the efficiency? Compare, I think the same question between the lysolecithin and the bile salt. This is the same question. Okay, move to what is the good source of bile acid or in this study? I think
I don't know, but I think the source of bile acid. So we can say like this: bile acid, lecithin, and lysolecithin. Lecithin, they have many functions in animal body, the same as bile acid. But some property about the promote muscle yield or fat deposit can also found, but not directly. But not directly. Generally, for the lysolecithin, we use it as firstly as the emulsifier, and also use it to be the we call it lipotope to carry the lipid from the hepatopancrease, prevent the fatty liver. Bile acid also can do, but not too clear on this function. Lysolecithin can be the good emulsifier, but it depends on the proportion of the fatty acid inside the molecule. So you need to select or concern the lysolecithin that contain high lecithin, high lysolecithin, better than another phospholipid. In fact, these 3 materials, the function very close together, and also can hold the other nutrient like the mineral or the vitamin to help the function of this material. Which one is better than another one, or which sort is good? I think it's difficult to say. It depends on the supplier. So you need to check with the composition of the material first, and then make the decision to do some trial Yeah, but bile acid, the high level of bile acid, I think is not too much effects on the on the growth performance or the bad condition. Lecithin also you can use it at a little bit high level. For lysolecithin, because of the molecule is modified, you need to concern or to check the optimum level to use this product, and also need some material to stabilize the emulsification condition.
Yeah, I fully agree with you. Every product has their own properties, own advantage and disadvantage. When we choose, we should think about it. Maybe we try first. So finally they can apply. Yeah, and then the last question, I think any stability concern when use bile acid.
Pardon, please say again.
Yeah, any stability concern when we use bile acid. That means maybe the virus, the bile acid not stable or stable. This is a question.
Bile acid generally is stable. It's not necessary to use any stabilizer, but if you want to improve the emulsification property of bile acid, yeah, add some more acid and a little bit mineral and monoglyceride or glycerol, maybe can prove. And also you come back to the 6 material that can boost the bile acid condition, like the taurine, glycine, choline, because of this one will promote the function of bile acid inside the animal body. Yeah, and also promote the efficacy of bile acid that you add it in as the exogenous bile acid. And also like the group of the methane, that can help the can be the lipotol and can be the osmoprotectant. Yeah, you improve the hepatopancrease condition. Yeah.
Okay, thank you. One more question, Prof. Orapint. Can bile acid improve feed with lack of nutrition? So that means maybe the feed lack of nutrition, then we add bile acid can improve the quality of the feed.
Just only bile acid can provide some cholesterol and phospholipid. In fish, maybe not too much significant, but in shrimp, I think if you add bile acid at a little amount, yeah, it can provide cholesterol and phospholipid, because of inside the bile acid cholesterol a little bit high, and cholesterol very important for shrimp and difficult to find the source, good source of cholesterol. Yeah, because of cholesterol very expensive. Yeah.
Okay, I think that's all for your question, and really big thanks to you for your patient answer so many questions. Okay, so I think thanks again for your presentation.
You're welcome, and thank you very much for the participant attention. Yeah.
It's our honor and our pleasure. You share your knowledge and some project to us. Yeah, I do hope our audience will learn a lot about the hepatopancrease. You know, that's healthy or unhealthy in terms of the photos, some index, all these things. So even I don't want to say this is the end of our webinar, but I have to say. So, all right, above all our presentation for this webinar. Now it's going to be the end of the webinar, and before closing, I would like to thank our sponsor, Shandong LachanceGroup, for organizing this insightful webinar. Appreciate to all our 8 speakers from Thailand, from Turkey, from Bangladesh, Indonesia, also from China. Of course, thanks for your sharing and also the effort for this webinar. And also, most especially, thank you to all our audience for joining us in the whole webinar. We do hope you have learned some new concepts, new ideas, and also bring home your learning that you can apply to your aquaculture business in future. And once again, thank you for attending, and we are looking forward for your participation in the future webinar. And stay safe. All the best. Thank you all.